5, 4, 3, 2, 1, 0. Iniciamos con el evento de nanomedicina. Bienvenidos a todos a este evento, hasta esta segunda sesión del evento del primer simposio de nanomedicina, biomateriales y bionanotecnología. Sean todos bienvenidos. Vamos a iniciar con una presentación del profesor Wouter Ross desde Groningen en vivo a través de Zoom. Y I'm going to now to switch to English, so we will start now with the first presentation. So I'm going to introduce the Dr. Professor Wouter Ross. So okay, so Professor Wouter Ross, he is, let me find his bio. He obtained his PhD in biophysics in Heidelberg, Germany, and after a postdoc period at the Max Planck Institute for Metalforschung in Stuttgart and an Institut Curie in Paris, he went to the Vrije Universiteit Amsterdam to focus on physical virology approaches. In 2015, Dr. Ross accepted the chair of molecular biophysics at the Zernike Institute at the University of Groningen. Dr. Ross, in that institute, in that lab, is heading a research group focusing on mechanics and dynamics from molecular to cellular length scales, including studies on viral self-assembly and mechanics, extracellular vesicles, and membrane proteins. By using techniques such as Highspeed AFM, optical tweezers, and fluorescence microscopy, his lab is aiming to describe and unravel physical principles of subcellular mechanics and dynamics, and to elucidate mechanisms behind protein and supramolecular assembly functionality. So, please, Wouter Ross, you can start with your presentation. Thank you and welcome.
Thank you very much, Armando. Can you hear me well? Yes? Okay, I heard some echo, but I think it's better now. Good.
Yes, we can hear you.
Okay. Thank you very much, Armando, for the nice introduction and for the invitation. It's really unfortunate I couldn't make it to Mexico, but I'm very happy to be able to join you online. And today I'd like to tell you a little bit about the biological dynamics and interactions we are measuring at a nanoscale using single molecule approaches here at the Zernike Institute in Groningen. We look at quite a few different systems, but I decided to focus this talk on viruses. Because viruses is the work we started with, and still a large part of the lab is working on viruses. So this is a very simple diagram of the viral life cycle. Biologists will not agree, they will think it's much too simple. But being a physicist, for us, it is quite useful. So even though not all viruses follow the scheme, and it's much more complicated than you see, a few key steps which undergo many viruses during the life cycle are in here. Well, first of all, we have attachment of the particles, then they enter into a cell, they encode, the genome is being replicated, so either new RNA or new DNA is being made, viral capsid proteins are being made, and together the genome and the capsid proteins assemble then into a particle, which is finally released. For quite a few years, I've been studying the mechanics of viral particles, so related to entry and uncoating using AFM. And now recently I also started looking at viral assembly, take a look how the subunits assemble and form a particle. So I want to discuss both things. I will start off with number one, with the mechanical properties of viruses. So let's use the AFM. Well, the AFM, we can get nanometer scale resolution, and conveniently we can do this in liquid. Next to imaging, we can also do force measurements, and those force measurements are very important in order to determine the mechanics of the particles. So how do we do this? How do we use AFM in order to study mechanics of viruses? I have a small animation over here. It includes sound, so I hope you will hear the sound. If not, I can try to explain what you see. But what you finally will see is an AFM tip scanning over the surface, looking at viruses, finding the viruses, and indenting them in order to find the mechanical properties. Okay, so that is the essence of the experiment we do. I hope you heard the voiceover, but as I said, if not, the essence is we look for the viruses and we start pushing on it. You see this in the schematic over here. We go to the center of the particle, we push hard on it, and we get our force deformation curves. Initially, we have reversible deformation. So from this part, we can then determine the spring constant. At a certain moment, the particles break, and from there we have the break force. So spring constant and break force are the two material properties with which we are characterizing our viral particles. And by changing pH, changing salt concentrations, but also maybe removing the genome, making a mutant, or adding some external proteins, we can take a look whether the mechanical properties are changing or whether they're staying the same. So we're looking at quite a few different viruses, and I'd like to give you one example, and it's of adenovirus. So adenovirus is an icosahedral virus. So almost more than half of all known viral particles possess icosahedral symmetry. So it means it's a 20-phased body with 12 vertices, with 12 corner points. And you see it here depicted in reconstruction along the 2-fold axis, the 3-fold axis, and the 5-fold axis. Well, we can also image this when they're lying on the surface. And here you see that they're sometimes lying on, they just attach randomly to the surface. Sometimes they lie on the 2-fold axis, and sometimes on the 3 or the 5-fold axis. It means if we start pushing on it, we push along one of these axes. Now, here's an image before indenting and after. This is an example of how we're pushing on the 3-fold symmetry side of the viral particle. And you see that the height of the particle decreased really a lot after indentation. Before indentation, it's around 80 to 90 nanometers, which is the diameter you expect for an adenovirus particle. After indentation, completely collapsed. And just like in an animation, where you saw that the particle shattered in small pieces, also here we see the particle shattered in small pieces. And those small pieces you can zoom into. If you do that, what you then see is that they are the hexons. There are 240 hexons in adenovirus. Well, we don't see 240 particles lying here because they lie all on top of each other. But if you take a look at the isolated particles, what we see is that they really fit the dimensions of the hexons as we expect, both the height and the width. If you take a look very careful, you see the 3 dots the free extensions you see over here, you also see it back on the particle. So the particle falls apart into its hexons. Of course, we're also recording the force distance curve in order to take a look at the mechanical properties. And we did it for the various orientations. Well, why did we do this? Well, in order to understand the mechanics better, but also to understand infectivity better. Well, our collaborator, Glenn Nemoff from Scripps Institute, already found out that, together with colleagues, that the adenoviral particles attaching to first a primary receptor on the cell surface and afterwards a secondary receptor, and that integrin, secondary receptor, then it's taken up via endocytosis into an endosome. And finally, it leaves the endosome. Also, the pentons, the penton base are being removed. And then this particle without endosome, so only the capsid, is being shuttled via microtubule-dynein-mediated transport towards the nucleus. And at the nuclear pore complex, then uncoating, and its DNA is being expelled into the nucleus, and then new genome and new capsid proteins can be made, and a new viral particle is assembled. In particular, what is known about the entry is that integrin is essential for DNA uncoating. It's not essential for entry, so the particles enter without integrin, but the DNA will not be uncoated, so the DNA stays inside the capsid. And furthermore, it was known that defensin, it's an antimicrobial peptide, is blocking adenovirus infection. What we did is we took soluble integrin and we added it to our viral particles and took a look what the changes in mechanical properties before we added the integrin, and after that we added the soluble integrin. In different experiments, we added human alpha defensin, and we take a look at what the difference was in mechanical properties before we added it and after we added it. Now, it was already known that integrin and defensin, they both bind at the 5-fold symmetry site at the penton base. And what we saw that after integrin binding, the penton stiffness went 50% down. And in a different experiment where we added defensin, the penton stiffness went 70% up. Now, this fitted what we were expecting, that there would be some mechanical change related to infection. The reconstructions of the group of Glenn Nemoff had shown that after integrin binding, due to some steric hindrance, because there are 5 RGD epitopes, but not all 5 of them can be bound. So only 4 of them can be bound by integrins, and due to the steric hindrance, there's some kind of twist of the penton base. And this twist of the penton base was seen as some kind of unscrewing, making it more flexible, so that the penton base could later be removed, as you see at number 4 of this schematic. Well, if you want to remove it, it needs to be softened. And exactly that's what we saw, that integrin is softening the penton base. And furthermore, somehow adding defensin leads to blocking of adenovirus. And here, the expectation, we didn't have a clear expectation of what would happen, but here we saw that defensin is increasing the penton stiffness, so it cannot uncoat. Well, this showed there was a direct correlation between infectivity and virus mechanics, and that was really exciting. Because when we started working with viruses in AFM, we were just pushing on them because we could push on it. We're physicists, we have a toy, we have the AFM, we can push on it, we can see how it behaves. Well, that was very interesting in the beginning. But then later, when we had the tool well established, we realized we can also try to understand biology better. And this is a very nice example of how we could see that there's this direct correlation between infectivity and mechanics. So, well, as I said, we've been looking at quite a few different viruses. Now, our colleagues all over the world have been looking at other viruses as well, taking a look at the mechanical structure. Also, many groups have been looking at the atomic structure of viral particles. And together, we have now quite a good view on mechanical and atomic structure of all sorts of different particles. Still, there are many open questions, so this research continues also in my lab and in other labs to better understand the mechanical properties, and for that we use the atomic structures which are available. So we know relatively well what a virus capsid looks like and how strong it is. Even though there are still open questions, we know already quite a lot. We know what it's consisting of protein, monomers, or dimers. What we really still don't understand is how it assembles. So this whole viral assembly is a fascinating but rather poorly understood self-assembly process. So therefore, next to taking a look at the mechanics of viral particles, I also want to take a look at the assembly of viral particles. Because conventional AFM is a very slow technique, it takes a couple of minutes to take an image, 3 minutes to 5 minutes to take a single image. It was really too slow to use for viral assembly studies. So therefore, I moved my focus on using optical tweezers in order to study viral assembly. Optical tweezers is a technique in which you can catch micron-sized particles in the focus of a laser beam. You see this over here. Developed in the 1980s by Arthur Ashkin. And if you want to know a little more, I can, for instance, refer you to a review by Bustamante and co-workers. Idea of optical tweezers is that you have a scattering force which pushes the particle away and a gradient force which pushes or pulls it towards the highest intensity up the gradient. And it is because of transfer of momentum from the refracted light rays. And if you have a high NA objective, then the refraction force is bigger than the reflection force, and you can stably trap a particle in the focus of a laser beam. You can catch one particle, but you can also catch two particles. If you just have two laser beams, you can catch two particles, and you can glue a DNA molecule in between. Now, if you have this DNA molecule, you can afterwards add proteins. So all sorts of proteins have been added to DNA molecules in optical tweezers. But viral proteins, capsid proteins, haven't been added yet. So that's what we tried. We tried to add capsid proteins and see what happens. But finally, what is happening during viral assembly is that a particle is packaging the viral genome in a small protein ball. Inside we have the genome. Outside you have the protein ball. So if you have separated the genome and the proteins, together, they should somehow assemble around the genome, making this capsid. So what we look is at the initiation of assembly. So we didn't look at formation of complete full infectious virions. We looked at the initiation of assembly. In order to do that, we could take a look at single-stranded and double-stranded genomes. Here you see an image of two beads. In between, there's a DNA strand. This is optical microscopy, and because the DNA is 2 nanometers in diameter, you can't see it. But if you move this particle, then you see that this particle follows a little bit, which means there's some invisible thread in between, and that's the DNA. So here you see removing this particle, but this particle follows a little bit, as you see over here. When the force is increasing, it's following a bit. Now we come to an overstretching regime where double-stranded DNA is melted and single-stranded DNA is being made. And this is what we see if we go back. We see there's a completely different behavior. Single-stranded DNA has been made. So therefore, we can choose whether we work on viruses that self-assemble around double-stranded genome or around single-stranded genome, because we can have both in our optical tweezers setup. But first, we start to take a look at synthetic virus-like particles to understand how rod-shaped artificial VLPs are being made. And for that, in fact, we used particles that were developed in Renko de Vries lab in Wageningen by Armando. You see here, he's first author on this paper, an excellent paper, and we were very excited when we saw this because we thought, oh, maybe we can do something with that. So we got in contact with Armando and Renko, and indeed, they were willing to provide us this biosynthetic code protein. It is a polypeptide with a random color-preventing aggregation, stacking the domain in order to have cooperative assembly, and the DNA binding domain in order that you have assembly around the genome. So if we mix the DNA and the polypeptide, rod-like particles are being formed. They appear rather stiff, and it is shown that the DNA is being compacted with an order of 3 times compacted, so the DNA length is decreasing from 100% to 30% roughly, and it's cooperative. So what we then did is that we added a fluorescent label to the polypeptide in order to see whether we can follow assembly live of a single particle. Well, and what you see if you add fluorescent label to proteins, you can see it binding to the DNA in the tweezers, and you can move it. You can see it moving around. So what we did, so over here we were trapping the beads with quite a high laser power. We added a fluorescent capsid protein, fluorescent polypeptide, and we took a chymograph. And the chymograph is just we take an image of the DNA, a line. We glue this here on top. And the next image we take of this line, we glue below. And like that, we glue all the images of a line of DNA below each other. So that's how you can visualize a movie in a single image. But what you see is that over time, that small fluorescent signals are appearing, which are diffusing over the surface, over the DNA. Also larger dots are appearing, and they stay put on the surface. They are not moving. Well, you can quantify this, because if you determine the fluorescence intensity of a single fluorophore, you can also you can just double that amount, the intensity of 2 fluorophores and 3 or 4, and that's how we could determine the oligomeric states. We see all sorts of oligomeric states are being added. And we could also take a look at the diffusion, because as I said, the big dots are not diffusing, but the small dots, the small traces are diffusing. What it turns out is that the larger the oligomer, the smaller the diffusion, and for oligomers bigger than 5, we don't see diffusion at all anymore. So small oligomers attach and diffuse, big oligomers are not like this. But wait, I was telling you I want to take a look at viral assembly. And here we don't see assembly because the B to B distance stays the same. I was trapping the beads with a high power, and the polypeptide doesn't have the power to really condense the DNA in order to make a viral particle. It attaches to the DNA, it diffuses over it, but it's not condensing it. So then we thought, well, instead of having this high laser power, let's go for low laser power. Let's see what then happens with the low laser power. And this is the following experiment. We're trapping it with only a low laser power. And what we saw over time is that the beads are indeed being pulled towards each other. So this is really live assembly of a single virus-like particle. So we were very excited about this because it hadn't been shown before. That you can really see live viral assembly. Well, and if you take a look at the fluorescence signal, you also see that a rod-like particle is being formed exactly as expected. And using a related technique, we could take a look at the steps of assembly, because here we have how the distance between the outer ends of the DNA is decreasing over time. If you zoom in, you can see that it's decreasing with discrete steps. Now, these steps are roughly plus or minus 30 nanometers, so we see packaging, but also sometimes a bit of unpackaging. We took a look at the viral particles in the AFM. We could determine their height, roughly 9 nanometer. And combining all the data, we could then conclude that the viral particle is assembling by wrapping the DNA around the silk core. And here around it, we have the random coil preventing aggregation. So together, we could show that we can really follow viral assembly at single particle level, and we're able to make a model of how viral assembly is occurring with steps of 30 nanometer compaction. Well, the reason I started studying optical tweezers, using optical tweezers to study viral assembly was, as I said, because atomic force microscopy is such a slow technique. And that's true, but in the meantime, another type of AFM appeared, that's high-speed AFM. High-speed AFM is much faster, so I thought, well, let's take a look if we can study viral assembly with high-speed AFM. First, I want to briefly explain what is high-speed AFM. It was developed by the group of Toshio Ando, and for some review papers, I would refer you, for instance, to these papers. And I think the publication that really shocked the bio-AFM world was the moving myosin motors on actin filaments. Toshio Ando showed that you can really see single molecular motors walking over an actin filament. I really had expected this impossible, but it was really possible. They could see it very nicely. Well, how was this possible? Why you first had to wait 3 to 5 minutes, and afterwards you could get 20 frames per second. But a lot of improvements which all had to be done in parallel, because only if you implement all of them, it works. First of all, for these very high speeds, you need very high resonance frequencies, and therefore low cantilever mass. It means ultra small cantilevers, still difficult to develop. You need fast actuators, and you use some dummy piezo and balance weight. Here you see the very small cantilever, and here you see the piezos with balance weight, and also dummy piezos on the other side, in order to keep the center of mass at the same position. Also need fast amplitude detectors and adaptive feedback. Well, it was assembled in such a system. Well, you don't need to know all the details. We have the cantilever over here. We needed they needed an objective in order to focus the laser beam at the backside of the cantilever. And then here we have the sample turned upside down and the piezos. Over here, the detector of the laser diode and the detector of the laser light, and a lot of electronics around it. Well, when we finally had such a system, we right away tried to take a look at viral assembly, but it didn't work out at all. So we tried a lot, it didn't work out. So finally, we first started with a couple of other systems. I will show you later some examples of that. Quite a few different systems to get the system really in our fingers. And when it worked, we went back again to viral assembly. And the first system we started was HIV assembly. So the virus responsible for AIDS. We see it over here in the schematic, where a mature viral particle has a capsid inside with a genome inside the capsid. Well, the capsid protein, here you see the capsid. The capsid is made up of the capsid protein, which assembles. Here you see the capsid protein, assembles in a hexamer. Many hexamers assemble. It's quite a large particle. So if you only look at the small part, it is roughly planar. And we could indeed see these hexameric shapes on the surface. So then we can also take a look at dynamics. So look at 2D assembly of these hexamers. You see how a 2D sheet is forming on the surface. Well, at least we tried it and it worked out. So we were really excited about that we were able to see viral assembly live occurring on the surface. So here you see all the capsid proteins adding and making big patches. There's a lot of analysis of it. What we see is that first we have a very low height on the surface, then we have an increase in height, and even later we have a further increase in height. So what seems to happen is that the monomers are attaching, lie flat on the surface. At a certain moment they hexamerize, and if they hexamerize, then they get bigger. We can take a look at small bits moving around the surface. Here's some trajectories of moving them around the surface. And typically when the small bits, the small yellow dots move around the surface, to the moment they attach to a growing lattice and they stay there. And you see snapshots of this growing lattice. So we're very excited that we could see assembly of a structure on the surface. Finally, however, we were not sure what is this really the hexameric substructure, yes or no. So we really had to zoom in order to see if those things which are moving around, which are growing, they're hexamers, yes or no, and whether we could follow that or whether we're just moving in too much, applying too much force, and we can't do anything. So we tried to zoom in to take a look at dynamics, and what we saw is that we can indeed follow viral assembly at a single molecule level over time. So what you see over here is that hexamers are falling apart. Here's one falling apart, here's one forming, here's again one forming, another one forming. So it's really a dynamic process of hexamers being created, but also disassembling. But it made us really very excited that we're able to see this in real time, assembly of such a viral capsid lattice. Here you see the hexamers, single hexamers. And we analyzed what was attaching, and we saw that monomers, dimers, and trimers are attaching. But it was also postulated that both monomers and dimers and trimers could attach. However, because we were imaging with 2 frames per second, we don't know whether those trimers are real trimers or whether they are maybe there's 3 monomers which attach very quickly after each other. within half a second, and therefore, with our 2 frames per second imaging speed, we can't distinguish between 3 monomers which very quickly attach after each other or a trimer. So therefore, at the moment, we're trying to increase the imaging speed. It should be possible to image an order of magnitude faster, and then we were able to say with more certainty whether those trimers are really trimers or whether they're just monomers or a monomer and a dimer attaching very quickly to each other. So that's work in progress. So hopefully next time we'll be able to tell you something about that. But what we could conclude is that it's really a stochastic process in which assembly is occurring. Also, this was already predicted, but it's never been shown that it really is such a stochastic process. There are many different pathways in which the hexamer can form, and also many different pathways in which these hexamers, or the dimers and trimers and the monomers, are able to form this lattice. Okay, so these were experiments on using the high-speed AFM. I previously showed you experiments using optical tweezers, so they both are for viral assembly. So then we thought, can we maybe combine it, study one system with tweezers and AFM? But for that, hepatitis B virus turned to be an excellent system. Hepatitis B virus is in a particle of a bit over 30 nanometer in diameter. The monomers readily assemble in a dimer, so you typically don't have 3 monomers, but you do have 3 dimers. And these 3 dimers assemble then into a capsid of 120 dimers. The capsid protein consists of an assembly domain, so which is important for the protein-protein interactions, and a C-terminal domain, which is important for the interactions with the genome. Well, we took our optical tweezers. Hepatitis B virus is assembling around a single-stranded genome. So instead of having a double-stranded genome with the VLP, we now have a single-stranded genome. Just by pushing hard on the double-stranded genome, we got our single-stranded genome. And we indeed see assembly. How do we know we see assembly? Again, as before, we have initially a large distance. Afterwards, we have a short distance between the beads. We can determine how the beads are being pulled together, and we see it being pulled together with discrete steps. Well, these discrete steps turn out to be steps of roughly 60 to 7 nucleotides. Again, we see packaging and unpackaging. And what we added was then fluorescence microscopy. With 2 different types of fluorescence microscopy. We added, we labeled the monomers fluorescently, or sorry, the dimers fluorescent. Well, we labeled the monomers fluorescently. Well, they form dimers, and the dimers were attaching to the surface, and they could diffuse over the surface. We also did another experiment. What we did over there was that we added an intercalating dye. What does an intercalating dye do? Intercalating dyes only attach to double-stranded genome. Well, we only had a single-stranded genome. So before we added capsid proteins, there was no signal at all from our cytox, from our intercalating dye. But after we added the capsid proteins, all of a sudden we saw dots appearing on the surface of the DNA, of our genome, of our single-stranded genome. But if dots are appearing on the single-stranded genome, it means that some double-stranded genomes being made, because only then the intercalating dye is binding and starts to fluoresce. These dots turn to be stable over time. So apparently the hepatitis B virus capsid protein is able to not only condense the genome, but also condense it so much that hybridization occurs and double-stranded pieces of the genome are occurring. And very nicely, so if you take a look at the cytox signal, that at the moment that we see an increase in cytox signal, so here's again a chymograph, the big green areas above and below are the beads. In between there's the DNA. At a certain moment, you see a signal appearing. You see it appearing over here. So it's in the intensity, you also see it clearly. And exactly at that moment, there's a decrease in end-to-end distance. So there's a clear correlation between the packaging step, where the beads are being pulled together, and genome, which means genome compaction, and the formation of double-stranded genome parts. So this shows that the capsid protein is really chaperoning formation of double-stranded genome. Well, furthermore, from those step size analysis, we could then determine that the capsid proteins can condense nucleic acid even in attention, roughly 11 piconewtons. Assembly footprint is roughly 70 nucleotides. And if you then take a look at the step size, taking into account the distance between the bases in a single-stranded genome, we could then determine that the work per condensation step is roughly 100 kBt. Well, this means roughly 1.4 kBt per nucleotide. If you take a look at the ATP-driven packaging motor of a bacteriophage, or a virus which infects bacteria, you see that is much bigger there. It's roughly 5 kBt, whereas for the hepatitis B virus, it's only 1.4 kBt per nucleotide. So it's a very small energy which is involved, but apparently large enough in order to compact the genome. Now, here you see the dimer. So taking all the optical tweezer data together, we could conclude the following. So here you see computed delta G's for different structures. Here we see a trimer of dimers. So this would be a delta G of minus 50 kT. Our tweezer experiment showed that we don't see this. So we don't see trimer formation occurring on the surface. For a regular pentamer, you'd expect minus 80 plus minus 10 kT. For a diamond-shaped pentamer, so also pentamer but different form, minus 90 plus minus 10. And for an hexamer, minus 100 plus minus 10. In fact, our tweezer data showed that either B, C, or D is occurring. So we could exclude A, that energy was too far off, but B, C, and D could occur. So we really dove again into our optical tweezer data in order to be able to say, well, is it now minus 80 or minus 90 or minus 100, in order to say which of those three things is the assembly nucleus. But our data was of very high resolution, we're very proud of it, but not high enough in order to answer this question. So then we turn to high-speed AFM. To solve, we added the capsid proteins to a surface. We saw very quickly assembly of higher order structures. And this is exactly what you're seeing over here in this movie, that higher order structures are being formed. First, briefly, dimers, and then, first briefly trimers, and afterwards even higher structures. Here's some snapshots of the formation of these structures, and a zoom in, and it turned out there were diamond-shaped pentamers and dodecamers. So the regular pentamer and the regular hexamer, they didn't form. We only had the diamond-shaped pentamers, and then two diamond-shaped pentamers together, and with two dimers formed this dodecamer. So this high-speed AFM imaging was crucial in order to further understand our optical tweezer data, and we were able to say that now viral assembly or protein-protein assembly is occurring via trimers and then diamond-shaped pentamers, and assembly around the genome is really the diamond-shaped pentamer with the nucleus, and you really need that in order to grow further. Combining all this data, we could say with the following assembly model you see over here, where trimers and diamond-shaped pentamers are being formed. And if you add the genome, it can grow further. Our high-speed AFM images or movies were performed on mica, which is negatively charged, which is a proxy for the negatively charged genome around which HPV is assembling. And then either these pentamers are grown by addition of dimers or by another diamond-shaped pentamer into a dodecamer. What you also saw is that we don't see these 2D lattices like in HIVs, probably because HIV is a very large virus with not a lot of curvature. Hepatitis B virus is a very small virus with a lot of curvature. So when a dodecamer is formed, you would in fact below want to add more dimers, but due to steric hindrance of the mica, that is not possible. All in all, with this we've shown how assembly initiation of hepatitis B virus occurs. I told you already a lot about viruses now, a lot of work we have been doing and we are currently doing. Just want to add a few more slides for those people who are not at all or not only interested in viruses. Because these single particle techniques are also very strong, not only pushing on viruses, but also pushing on vesicles, on liposomes or extracellular vesicles or lipoprotein particles. Tweezers are very strong in order not only to look at DNA-protein interactions, but also at complete cells. Here you see work done together with Patrick van Rijn, who you also will hear speaking on single cell activation kinetics, also very exciting work which we use the tweezers. And also the Highspeed AFM, I already told you, viral assembly didn't work out in the beginning for us, so we turned our attention to some other systems to get our fingers wet. Then we could very nicely show how escort systems are responsible for pinching of vesicles, how that works, or at least some subcomponents, how that works. We can take a look at transporter systems, which are moving up and down in the membrane of bacteria. We looked at assembly of synthetic polymers, different types of polymers we could see growing on the surface. And very recently, we also started with antibiotics, which are very nice results, and which you continue to work on quite a lot now, taking a look at interactions of antibiotics with a membrane and how it is disturbing the membrane, and that's how it's leading to the killing of bacteria. So the techniques I showed are not only very strong for viral studies, but also for all sorts of other studies. We keep on doing these kind of studies. So, okay, well, with that, I would like to come to the acknowledgements. The work was done by current lab members, which you see over here, by former lab members, which you see over here. Our collaborators are essential, and of course, our funding. So, with that, I want to finish my presentation. I'll just keep on this slide, and if you have any questions, I would be very happy to answer them.
Thank you very much, Wouter. So now we will start with the session of questions. So please, if someone has a question, either here in the library or in the auditorium, just let us know. Raise your hand, please, and we will give you the microphone. Okay, one here in front.
Hi, Wouter, Patrick here. So excellent. It's always nice to see your results. I was wondering, so you looked at the two beads with the DNA strand in there, and then you could see the beads going back together, showing that you had an assembly around the DNA strand. Did you also then pull it back apart to see whether the force can also disassemble it again?
Yeah, thanks for the compliment. Yeah, indeed, we did it as well. So, and what we saw is for certain viruses, it seemed that we had complete assembly. For hepatitis B, we didn't see this. For others, it looked like we had complete assembly, and there we could not pull it apart anymore. But if it was only assembly initiation, we could indeed see we could pull it apart. We saw a lot of drops in the force distance curve, so they were clearly popping off. But interestingly enough, when we then, if we did this in a buffer channel, so there were no free proteins there anymore. If we then pulled again on it, so we pulled on it, we let it relax and pulled again. We saw again a similar kind of behavior. So apparently the bonds were broken, but the protein was still remained attached to it. And when we were relaxing, it was again packaging it. So we could do it then repeatedly. So indeed, for the assembly initiation, we could do it. For complete viral assembly, there it was just too strong. We couldn't break it apart anymore.
So does that mean that you could also dynamically study the stability based on the environment? So I can imagine that if you have high salt, that it destabilizes or becomes more stabilized.
Yeah.
So did you ever try to change the surrounding conditions to measure this?
Yeah, no, we haven't. It is already very challenging to do it like this with the different channels with different buffer solutions. It would be very nice to try this. For the moment, we've mainly been focusing on optimizing our system and getting enough data on this, and it's a challenge. But I think it would indeed be very nice to try this, to see how it is changing.
All right, thanks a lot.
Thank you.
Okay, so more questions? Yeah, one more there.
Thank you so much for the talk. Just wondering if the AFM nanoindentation also works on bacteriophages to see their infectivity, or only on capsid viruses.
Yeah, no, indeed. You can also do it on bacteriophages. We also did this. We looked at phage lambda, for instance, and phage HK97. And there, indeed, you can very nicely see on HK97, it's a particle which undergoes maturation. And there we look at the procapsid before maturation and at the capsid after maturation. You can artificially blow the capsid up, exactly what happens during maturation, but you can trigger this just chemically. Yeah. And you can see how the capsid is being reinforced after this maturation transition. Covalent crosslinks are being formed between the capsid proteins, really making the particle much stronger. So even though it is bigger, it has a thinner shell, it has a larger diameter, it still gets stronger due to these covalent crosslinks that are inserted in maturation. So it's a very nice approach to take a look at bacteriophages as well. Yeah.
Okay, thank you.
We have time for one more question. Anyone or a comment? Remember, there are no silly questions. You can ask anything, don't worry.
Indeed,
or in Spanish also if you want. Okay, well, if not, thank you very much, Wouter, for this nice talk.
Okay, it was a pleasure to be there and enjoy the rest of the conference and all talks. Thank you.
Thank you, and hopefully see you in Mexico soon.
Exactly, I hope as well. Okay, bye-bye.
Bye. Okay, thank you very much for these sessions. So we will continue with the event. Today, we will start actually with the presentations here with the presenters. So I'm glad to introduce Dr. Patrick Van Rijn. He will give us a talk about nanogels. So it's very, very interesting topic. And so I will say something about Patrick. Professor Patrick works in the general field of materiobiology and nanobiomaterials. Dr. Van Rijn has research interest in developing new dynamic biointerfaces. So that is one of the keywords, biointerfaces, for his talk, in order to understand the relationship between material properties and cellular behavior. For this purpose, his group has developed high throughput technologies to investigate surface parameter-dependent biological phenomena, but also have created novel coating approaches and delivery strategies using nanohydrogens. His research is conducted in both fundamental and applied character, with strong connections to the clinic and industry. I have to say that Dr. Patrick Van Rijn is associate professor at the University of Groningen and University Medical Center of Groningen at the Department of Biomedical Engineering, but also he is a founder and a chief scientific officer of an enterprise called BioMACS, which, okay, well, he will say exactly how it's, but this is a clear example of how he can combine lab research with application in a company. So just to finish his introduction, Patrick has been the chair of the Netherlands Society for Biomaterials and Tissue Engineering, program leader of the research program Nanobiotechnology and Advanced Therapeutic Materials, topical editor biomedical polymer for the open access journal Polymers MPDI from the MPDI publishers and guest editor for Polymers, Bioactive Materials, and Tissue Engineering A, which are very nice journals in this topic. Okay, so please give an applause to Dr. Padre Barbay.
Right, thanks a lot for this nice introduction. I hope everybody can hear me. So for those who yesterday attended Theo's lecture, so half of my group is basically looking at bio interfaces more from the fundamental point of view and how do materials deal with cells. And we translate that to medical implants, and that is also why we founded a company called Biomax. Today, I'm not going to talk about that because I'm going to talk about what the other half of my group is doing. And that is basically talking about nanogels. And nanogels is something that I learned during my postdoc in Germany in the group of Alexander Beuker. And while you pick up things and systems, I did not work with nanogels myself during my postdoc, but I was so fascinated about the elegance and the simplicity of the system that I had to continue with that. So today I would like to discuss nanogels. I just have to, yeah. So nanogels, and I hope that I can convince you, or at least instruct you that these are really an indispensable nanobiomaterial tool for nanomedicine. So tomorrow I will elaborate a little bit more on this, but today I will just stick to the highlights. But before talking about nanogels, sorry, I'm not an engineer, so this is beyond me. Yeah. So if we're talking about nanogels, I would like to start with something that can draw a little bit of inspiration, and that is basically the sponge. So if I look at a nanogel, I think of a sponge, and a sponge is actually a wonderful thing. We all have one at home. Well, the video doesn't play anymore, so that is a bit of pity. But I think you can all realize that if you have a very dry sponge, you put it in water, it swells up. The nice thing is that if you then squeeze the sponge, everything that was soaked up by the sponge will be liberated. So that is basically technology is not working on Tuesday mornings. But still playing the movie. But the nice thing, and if you look at the movie, then you will see that it soaks up water. If you squeeze it, of course, all the water goes out. But if you then put it again in water, then of course it takes it up. So this is really a reversible process. And it's of course interesting because imagine if you use your sponge to take up some dirt, you really want to also be able to get the dirt out. But you also would like to get stuff taken in. So this reversible process is actually very useful. So if I look at the sponge, so I always like to give a little bit of a definition to things. So the sponge is actually characterized by readily absorbing water and becoming soft when it does, but it retains its shape, its toughness, so it does not fall apart. And I think that is particularly interesting. So if you would remove the water, the particle is still a particle. And there are many systems that are excellent, but if you remove the water, they don't work anymore. So here are a couple of examples of sponges. As you can see here, this is the favorite sponge of my kids. But it is very diverse. And with diverse, so this is already what I showed you. So it can take up water, but not only water, it can also take up more complex fluids. So here you can see a sponge that has taken up blood. So blood is quite a complex mixture. And it can also reversibly release the blood again. Like I said, so a sponge is really, or the sponge can really be a solid object. So that also means that if you apply force to it, it still retains its shape. you can use it to, oh, I'm going to go a little bit too fast. So you can use it actually to dispense while applying a force without it breaking. And here, you can also appreciate this, is that a sponge is, of course, a microporous system. So that means that certain things can go through, but if the object is too large, it cannot go through. So it can also act as a filtration device. All right, so of course, I'm not here to talk about sponges that we use in our daily lives. I'm going to talk about sponges that are basically nanosponges. But in my mind, the same principle holds. And of course, it does not comply on a physical level. So if we would argue with a physicist, then of course the principles in how water is being taken up is completely different. But the concept of having a porous network that is very hydrophilic, can take up water, it holds also onto the micro and the nano scale. You can very nicely see this here. So this is a microgel. You can really see that there is a lot of porosity there. It is one big structure. You can really envision that these pores can take up water just like this sponge. If you go really, really small in the nanometer regime, then of course we lose a little bit of detail, but still the same holds. All right, so what is so special about these nanosponges? So these nanosponges, we call them nanogels, because in fact they are gels. But they have very peculiar behavior. So they behave in essential in 3 different ways. So you can compare them to macromolecules, because the nanogel is one complete covalently bound network, just like a macromolecule. So it is only one structure, and it is all connected. Additionally, it is also a particle, so it behaves like a colloid, and it also behaves like a surfactant, because like similar as that Theo discussed yesterday with protein adsorption and interfaces, that happens with colloids as well, and colloids can be used to stabilize interfaces, just as surfactants can. So this particle has 3 excellent opportunities to be used. It is completely cross-linked. And it's swollen by solvent. So if you choose the appropriate solvent, it really swells up. You can control this. The swelling is influenced by its environment, and we come to that a little bit later, because you can make fantastic responsive materials out of these nanogels. And if you look at the surface, and the next slide looks at it a little bit better, it is not a uniform particle in the sense, it is a uniform particle, but there is distribution in density, at least in the way that we prepare it. So in our case, we have quite a dense core, and it extends in a gradient fashion outward. So the core is slightly denser than the outside. So the outside is really, really fuzzy. So you still have dangling polymer chains, but they are part of the network. If we're talking about responsiveness, so a network that is able to respond to its environment, we can think of temperature, pH, ionic strength, but also light. And so all these compositions, we already saw them also in the talk by Helder and by Ali. So these are very useful properties that you can control and also utilize within the body. If we're talking about responsiveness, then what do we actually measure and what do we see? So here you see a curve where the polymer is, or the nanogel is responsive to temperature. So on the x-axis you have your temperature. Here we measure the hydrodynamic radius. And what you can see is that this polymer has a phase transition temperature of around, I cannot really see it here, but I guess it's about 30 degrees because it's NIPAM. And what you can see is that the hydrodynamic radius is decreasing to above the phase transition temperature, and then it is stable. But that means that during this process, it expels all the water. So the nanogel shrinks, it expels all the water, and everything that is in it, which is not bound, and is small enough to penetrate the mesh, and is reversible. So if you then just cool it down, then it expands again. And you can do this with temperature, but you can also do this with pH or with ionic strength or UV light. All right, so there are a couple of different ways how we can synthesize these. And so we can make use of emulsions, where we actually just make a emulsion system. This can be a macro emulsion or a mini emulsion. This can be oil and water, water and oil. In the end, particularly for the emulsions, you need to isolate the particles, freeze dry it, and then resuspend it, because here it is in a collapsed phase, and then only when you transfer it back to an aqueous phase, it swells up again. You also have dispersion polymerization, and actually this is our favorite, so the bottom one, and this is also why I highlight it, because this is our preferred synthetic method. It is very easy, it's very straightforward, because basically what we do there, and we'll explain it a little bit here, is we just take two monomers. Here you see NIPAM, and NIPAM is a thermoresponsive polymer. The key aspect here is that the monomer is not thermoresponsive. So it will only become thermoresponsive when the polymer is long enough. And of course, we need something to hold the network together. And here you can see a bis acrylate, so that has two functionalities that can both react. So by that, you can couple chains. So what we do is we add an initiator and we heat everything above the phase transition temperature. And this is actually crucial to form these kinds of nanogels, because what happens is that you have the initiator. So the initiator is this peroxide that homolytically cleaves into a free radical. And this free radical is actually starting a free radical polymerization with the acrylates. So these monomers, they start reacting and they form oligomers. These oligomers, so at the end, there's still an active radical and they become longer and longer. NIPAM, and as with it, most of the responsive polymers, they only become responsive when the polymer chain becomes long enough. At that point, the thermoresponsiveness kicks in, and you can see that it collapses on itself and it becomes a colloid. But it does not stop growing. So still there are active radicals on the surface and inside this colloid. Of course, there is still free monomer floating around, so the reaction is still continuing. Of course, you need to add a stabilizer there. At a certain point, the reaction is finished. If you then change the temperature, so you don't have to dry it and transfer it, no, you just set up the temperature, you wait until it cools down, and then what you find is that you have really nicely hydrated particles. So this volume phase transition temperature, so we do the synthesis above this temperature to set in this thermal responsiveness, that allows us to precipitate, and hence it's called precipitation polymerization. So this is this VPTT is also denoted as the lower critical solution temperature. So that's basically the temperature at which the polymer starts to precipitate. And what really happens is that you have nicely hydrated polymers in the hydrophilic state. And once you change the microenvironment, and that of course depends on the type of monomer that you choose. So in the case of NIPAM, it is temperature. So here you would increase the temperature. And what it does, all the bound water is actually liberated and the polymer collapses on itself. And this is completely reversible. All right, so that is a little bit of a quick background on nanogels. There is of course a lot more to tell, but I would like to keep that for tomorrow. So this is basically a tool, and this slide should make you appreciate the things that we can do with these nanogels. Because you can envision that depending on the monomer, there are literally thousands of commercially available acrylates. So if you go to Sigma or whatever, you can have a whole list to choose from. So it's really easy to actually control the chemical functionality. Because during your synthesis, you can just add other types of monomers. Additionally, if you look at the mechanical properties, we use a crosslinker, a bis acrylate. So the ratio of the monomer versus the crosslinker dictates how stiff or how soft a hydrogel becomes. That works also on a macroscopic level. The same holds on the microscopic and the nanoscopic level. Additionally, you can use not only specific monomers, but also combination of monomers to actually tune the range of your responsiveness. So NIPAM is 32, NIPAM is 45 in temperature, but you can also make copolymers out of them, and you can actually tune the temperature at which this kicks in. And this is actually also the very funny part, is that so normally you would make homogeneous nanogels. So in terms of that, the chemistry is distributed in a random fashion. We can also make core shell, so that we first make one core of one monomer, and then we add a second monomer a little bit later, so that it only resides in the shell. This is something that we personally don't do, but it is available, where you actually make Janus-like structures, so that you basically have your nanogel, you cut it right in half, and the left half has different properties than the right half. And so you can make excellent systems with this. You can all control this. So imagine all the possible combinations that you can achieve with this. All right, so how do we apply it? Because that is, of course, ultimately what you would like to do, also apply it. So you can envision that you can load the nanogels, loading it with drugs, for instance. You can also make them active. So you can build in components that actually interact directly with other systems. And in our case, that is, of course, cells. But although these are in suspension, we can also apply them as coatings. It's very straightforward. It's very easy. They're very stable. So this is a little bit the playing field. And if you want to really know more details, then I would refer you to this very nice review. All right, so I would like to highlight a couple of systems that we do. So we do it both on surfaces as coatings, but also intracellular mammalian cells, but also interaction with bacteria. I cannot discuss them all right now, so I will go in more detail tomorrow also on those systems. But I would like to highlight these three systems because they provide us with a general idea on what we can do and how it is important. So what I would like to start out with is discussing the mechanical properties. And not only the mechanical properties, but actually the physical-chemical properties. So how can we control the physical-chemical properties? This is simply by changing the density, so we can add more or less crosslinker. And that, so if we add more crosslinker, it becomes more stiff, it is less deformable, the density is higher, so we can really change this by very simply changing this ratio. Also, so you can see that here, this is not charged. So the NEPAM is not charged, this is not charged. But depending on the initiator that we choose, we can make the surface charged. Because if we have a positively charged or a negatively charged initiator, then the surface of the nanogel becomes negatively or positively charged. Because once it collapses, so the initiator initiates the polymerization, the polymer collapses, but the initiator, the first molecule that triggered it, is hydrophilic. So that will reside during the polymerization on the outside. So it will grow, but the initiator will remain. So if you use neutral monomers, then the initiator will take the upper hand in delivering the charge. But also just the chemistry, taking mixtures of monomers. You can build in really a lot of functionality in there. And I will show you a couple of examples. So I would like to start out by explaining the mechanical properties. So this is something that we find fascinating. So not only for nanogels, but also for our microscopic interactions with cells. And what we did was just making three different nanogels. Very simple. And of course, our aim was to keep the hydrodynamic radius, or in this case, the hydrodynamic diameter, quite similar. Because if you're looking at something, whether it's cellular uptake or anti-fouling capabilities that we're going to discuss now, then you would like to have only one parameter that is a variable. So here we thought, okay, let's keep the particle exactly the same. So the starting particle is in all cases about 100, 110. And if we then do the heating ramp, yes, we gradually change the temperature, we increase it, and then we can see that all the curves, all the particles, they actually collapse. But they do not collapse all in the same way. What you can see here is that if you look at the blue dots, the blue dot here is only 1.5 mole % of crosslinker. You can see that the difference between the before the phase transition temperature and after, the difference in size is a lot more than if you go for the highly cross-linked particle. And this is basically the deformability. So you change the stiffness, but you also change the deformability. Well, of course, we characterize this quite well. Here you see the relative cross-linking ratio. You can see that in all cases, they become negatively charged because of the negative initiator that we use. We measured the elastic modulus. We did this with AFM. You can see that an increased crosslinker will increase the mechanical properties, going from 20 to about 350 kilopascal. Very nice. This coincides very nicely with the deformability. So this is the deformability. And so this is basically the swelling ratio. And if you then also want to image that you have a particle, so what we can do is we just put it on the surface and analyze it with AFM, which we did anyway, because that is how we get the elastic modulus. And what we can see is that also on the surface, particularly we just focus on the hydrated surface, we can see that the height of the particle is very, very thin, much thinner than what we see for its radius. And if we increase the cross-linking density, we can see that the deformability becomes less. So that is in solution the case, but also at the interface. All right, so what we did then is we applied this as a coating. That is something that we do quite often. We got this technique, so we did not develop the technique ourselves. We got it from a group in Potsdam. But we extended it to all known materials. So what we did here was we took a broad range of materials, ranging from elastomers, polymers, to metals, glass. And what we did was very simple. We just plasma oxidized it. We found that if you plasma oxidize a surface, doesn't matter what surface it is, it always becomes negatively charged. In our case, of course, our particles are negatively charged, so we do need one procedure in between. So we dip it in polyethylene imine, so that you have basically a layer-by-layer deposition of polyelectrolytes, renders our surface positive, and then we just spray our nanogel on top. We wash it, so just as a layer-by-layer deposition, there is no interaction with a negatively charged particle and negatively charged particles. We can wash everything away. And it doesn't really matter what kind of material we use. We always get these nice little densely packed coatings on top of our material. So if we zoom in, so this is how it looks like for the low modulus. This is for the intermediate modulus, and this is for the high modulus. So you can see that it is composed all out of spheres. The surface is relatively flat, so it's really, the roughness is about 8 nanometers, 11 nanometers, so it's really small. And what we did was looking at bacterial adhesion, because we know that polymer brushes, they are excellent for anti-fouling capabilities. Bulk hydrogels, if they are dense enough, and they're also excellent in anti-fouling capabilities. But for nanogel coatings, this was not really known. And since we had nicely the capabilities of changing the physical-chemical properties of these nanogels, we really thought, okay, this is something interesting to look at. So what we did is we took glass as our substrate. We coated the glass with either the low modulus or the intermediate and the high modulus. And we just put the coated surfaces and the control surfaces in a flow chamber. We suspend the flow of bacteria across it, and we just count. And we just count the number of bacteria that are adhering. And if you then quantify that, then you can see that, and this is in log units, so that it is more than 2 log units decreasing if you look at the control. So that means that here the soft nanogel particles and the stiff nanogel particles, they all have the same anti-fouling capabilities. That was something that we did not anticipate, because we thought, okay, the mechanical properties, the deformability, that should have an influence on the adhesion. So these are excellent results. I mean, this represents about 99.2, 99.5% reduction in bacterial adhesion. But we were a little bit bothered by the fact that we did not find any differences. Then we started thinking, oh, a little bit too fast. If you want to compare something, then you need to compare it properly. And while we kept a lot, so we did a lot of synthetic efforts in order to keep the hydrodynamic radius the same, as soon as you put it on the surface, and that is of course where we were interested in, on the surface you get different coating thickness. And that is of course not a fair comparison, because if a bacteria comes at the surface, it needs to indent a little bit the hydrogel layer. If it indents it 10 nanometers, then it will already see the subphase, and then it thinks it's on a solid surface, so it will adhere. So the fact that they did it, but that there is no real difference, we said, okay, we should do something different. So we did another set of experiments where we said, okay, now we're not going to make the hydrogel or the nanogels the same size in suspension, we aim for to achieve the same height when we apply it as a coating. So we tailored the hydrodynamic radius in such a way that if we apply it as a coating, we get the same coating thickness. Then we have a fair comparison. We did the same experiment again. We have a flow of bacteria across our substrate. And what we then saw was that indeed, the softer particles with the same coating thickness are in fact more efficient. It is not a lot, so it saves a couple of tens of percentages, but still, what we could show here is that, in fact, it's not only the mechanical properties, but also the thickness of the coating of the nanogel that does it. So bacterial adhesion decreases with lower cross-linking density, but it also decreases with a thicker coating. Well, we were quite happy because this actually said, okay, we have an approach where we can apply these nanogels as coating, and also our mechanical properties, we can tune it and we can apply it for something useful. So this is at an interface with bacteria, but of course we're not only interested in bacteria, we're also interested in mammalian cells. And then I was discussing with a colleague of mine, Inge Zuhorn, who's also associate professor in nanomedicine, and she looks specifically at the blood-brain barrier. And we knew already that softer particles were internalized easier in Hep G2 cells. So they are taken up faster and in larger amounts. That is what you can see here. So here you can see the increase in cross-linking density. So in this case, they used HEMA bis, but you can see that the percentages are quite similar as that we used. And they already knew that if you increase the cross-linking density, then you have more uptake and it goes faster. So that was our starting point. So if you sorry, I have to say it correctly. So softer particles, they were internalized more. So decreasing the stiffness means increasing the uptake. And there are quite a lot of mechanisms where particles can be taken up. So we did not study all these mechanisms. But since Inge was very interested in the blood-brain barrier, you have to appreciate that that is, of course, a more complicated system. Because if you're looking at the blood-brain barrier, it is actually composed out of three phases. So first, the nanogel needs to interact with the cell membrane. It needs to be taken up by the cell, so it needs to go from the blood side to the intracellular side. But then it's not done yet. So in order to become transported to the brain, it also needs to be excreted to the brain side. And that is the transcytosis process. So we thought, okay, we have different types of nanogels with different mechanical properties. We know the mechanical properties are influencing these processes. So we redid the synthesis, but now we added the fluorescent label so that we can also detect what is happening to these nanogels. We again altered the cross-linking density, and of the middle cross-linking density, the intermediate cross-linking density, we also changed the size, because we also know that size matters. And then we looked at the different stages of nanogels being taken up, in this case by CME-C3 cells. We applied it as a monolayer, so Inge devised a new blood-brain barrier model, not on transwell plates, but based on a collagen hydrogel layer with on top of that the endothelial layer in a closed packed system, and then she can isolate all the different phases. So it's a very elegant system. And then what we looked at was nanogel uptake. We did it at two temperatures, so at 4 degrees C and 37 degrees C, because what 4 degrees C does is basically switch off all the uptake mechanisms. So then you're only looking at adherence of the nanogels on top of the surface of the cells. And at 37 degree, of course, you have both processes. And what we could see was, in fact, that if we look at the fluorescence intensity per cell, that there are more particles, more nanogels being taken up by the cells if they are stiffer than when they are softer. Also, if you look at, so that was one surprise, because in the previous studies, they showed that softer particles are being taken up more. But also, to our surprise, was that if we apply a particle of about 200 nanometer or a particle of about 500 nanometer, the uptake is the same. So in this case, size does not matter. So that was very interesting because this already deviated from what we thought, right? So if you find something that is different than the established literature, it's always interesting. So what we in fact found was that an increasing stiffness means an increasing uptake. All right, but then of course we only have part of the story, because transcytosis means that you go from the blood side to the brain side. So you're not only looking at the cellular uptake, you're also looking at excretion. So that is also what we did. So we analyzed, okay, how many particles are in the cell layer and how many particles, what is the percentage of particles that we can actually find in the collagen layer underneath the cells. And again, to our surprise, what we found was that the lowest percentage was for the most stiff nanogels, while the soft nanogels were higher. So if you correct this, so for uptake and transcytosis, so how many are actually at the brain side, and you correct for this, then you can see that it's almost a factor 2 difference. And it is more striking because we have more uptake by the cell. So if you have more uptake by the cell, you would expect that more particles are also being excreted to the other side. But in fact, that is not the case. Right? So even though the soft particles are being taken up less efficiently, it means that once they are being taken up, they are extremely efficient in going to the other side. And this low uptake, with the combination of still high transcytosis, is actually a new way of how we were now starting to look at different particles. And Inge had a major grant on this idea, because what we came to realize is that, hey, if we can combine these two, so why don't we try to design a particle that is stiff in the beginning, so actually have a high uptake, but in that process become softer? So can we utilize chemistry to make that happen? Well, we're not that far yet, but this is a different way of looking at nanomedicine, right? So not only in which barriers, but also should we not have particles that change their properties while they are crossing all these barriers, because they're every time in a different microenvironment. All right, so of course now I only did, I will speed up a little bit. So I talked about mechanical properties, but I did not discuss anything useful, right? In terms of, okay, can we actually deliver something? So what would be a strategy? Because we already heard liposomes, we already heard lipid particles, or silicon particles, and so Helder already gave a good introduction to that. But what about nanogels? How do we deal with this? So do we supply hydrophobic drugs or hydrophilic drugs? So one way that we thought of was, hey, maybe we can utilize the stability of bonds that we add in our network. So we can quite easily make new monomers. So these are actually commercially available, but making new monomers is not that difficult. So here our approach was, we take an ester, we take an amide. We know that esters hydrolyze more easily than amides. We stuff them in a nanogel. So we have one nanogel with ester bonds and one nanogel with amide bonds connected to the fluorophore. So a labile nanogel and a stable nanogel. So what we would expect, is that if we introduce then the labile nanogel, so with a labile connection, to the intracellular environment where there are a lot of esterases, this bond would be cleaved, and then you would have diffusion of your dye inside your cell. While if you have an amide gel, which is more stable, it would remain punctuated. Well, this is what we did. So here we used so the red picture is the now blue. So here is the amide conjugated dye. This is the ester conjugated dye, so the FITC. And what we indeed see, when we add this, we see a very diffusive green color. Right? So that means that the dye is no longer solely connected to the nanoparticle, to the nanogel. Well, if we apply the stable dye conjugated system, it remains punctuated. So these are just two separate experiments. Then we thought, okay, maybe there is variation in the system. So why don't we just add both particles to the same system and look whether we can identify the same results? If we then look again at the now blue, so the red fluorescence, then we can still see this punctuated system. If we look at the green, we see a very diffusive system. So these are really in the same cells. You can see that the cell pattern is also exactly the same. So here we zoom in, you can still see that the red dots are punctuated while the green is very diffusive. So this means that indeed we make use of the intercellular environment, and we also, of course, tested this and confirmed that this is based on the intercellular environment. We'll skip that, but of course the proof in the pudding is when you combine both the stable and the labile in the same particle. So in the previous experiment, we just did a mixture of particles. So now we designed them both in the same, we applied it again, and we basically get the same result. So really that we can functionalize a nanogel using a tracer that does not come off the nanogel, so we can trace it. And then on the other hand, we can conjugate something by an ester bond, and then it is liberated intracellular. So of course we can do more functionalization on the nanogels. That is a little bit what I'm going to discuss then tomorrow, but also to actively interact with biological systems and deliver hydrophobic drugs, because this is of course a hydrophilic component, and you can also easily imagine that we don't use a fluorescent dye, but a conjugated hydrophilic drug. So we are also expanding this to other systems and hydrophobic systems. But I hope that I've shown you that a nanogel is a really versatile particle. You can do quite a lot with it, good control on the physical chemical properties, and really apply it to the functional system. Obviously, I'm not going to say that this is the ultimate system. I think for every system you need to tune it. And this below, I just want to highlight a work of a friend of mine, Felix Plamper, who's in Germany, where he actually used liposomes, which are well known. He decorated the nanogel with these and then increased the temperature. You can see that the nanogel is shrinking, but of course the vesicles not. But you create pressure on the vesicles and they burst open. So the combinations of all these systems, and I think that is where the strength will lie in the future, where we utilize combinations of systems in order to achieve something useful. Right, so these are all the people, and the ones that are highlighted, those are the work that I highlighted today. Thank you for being here, and if there are any questions, I'm free and happy to answer.
Thank you for your very illustrative talk, very understandable. I just want to ask about the molecular weight ranges of the nanogels.
That is a very good question. So in principle, you cannot really do proper molecular weight. So they are way too big to detect with. They are way too big to detect with mass spec. So in principle, what you can do is make an estimate on, okay, how much is the dry weight of a number of particles, you know the dimensions of the particles, and then you can make an estimation. I would not dare to make an estimation, because that is also for us not really a main question on how heavy they are. It's more, okay, what is the size? Because the size for cellular uptake and those things, those are more important. But it's very difficult to estimate a proper molecular weight of these.
Hi, thank you very much for the talk. It's really interesting, the applications that these nanogels have. My question is regarding the assays that you were trying to compare the stiffness of these two nanogels and how they interact with membranes, with cellular membranes. I was thinking, I'm studying the bachelor in bioengineering, and in our lab, we are trying to measure interactions between with particles and with a lipid monolayer as a model of a cellular membrane. Could this be maybe a strategy to actually compare if the stiffness changes how it interacts with the cellular membrane, but with a model membrane, not with a complete cell? That's my question.
Yeah. That is a very good question, and luckily Wouter was already ahead of me, because that is going to be something in the future, right? So Wouter has a very good way of looking at nanoparticles, how they deform, measuring the mechanical properties, not only of the particle itself, but also of the membrane itself. So I think combining these high resolution or high speed AFM together with planar membranes, and then just applying these different nanogels to them, and then see how do they deform at the interface. So that is definitely going to give us more insight in how they actually are being taken up. And in fact, I think the extension to real mammalian cells is not that far, because also there we are already able to image the surface of the cells. We can determine mechanical properties of the cells with AFM. So I think that connection there is very interesting to exploit.
Thank you, Patrick, for a wonderful talk. And I think after looking at your talk, my mind went back to something we have discussed before. Because yesterday I was telling everyone about these molecules, if they sit on a surface, hold on to water, can lubricate the surface, decrease friction. Now, your coating seems to do the same. It holds on to water. Do you think it can also give rise to a very lubricating coating?
Yes, but I don't think the Nippon coatings. I think you need to have a balance, because, of course, if you if you apply pressure, that is also how antifouling is working, right? So if a bacteria comes, it needs to compress the layer. If there is not enough energy to actually do this, it will not do this, because all the water needs to be liberated. So once you are applying force to it, so whether you do force as an energy or temperature as an energy, it doesn't really matter. You are able to push it. So you actually push. So if the force you apply is large enough to dehydrate your polymer, you will probably not find a difference in friction, unless you have a system. And so now you're asking the question, and then my mind goes to switch ionic nanogels, which we are now doing as well. So I think we should repeat some of our previous experiments, because then you have a system where water is really bound. Yeah, so here you make use of the reversible binding of water, because that is what it makes it responsive. But if you go for a system where water is really tightly bound, then you might prevent this decompression, and then you're talking about different things. So then it might work.
Okay, so you did think about our results before, and
Of
now
course.
Very good. Thank you very much.
Is there any question in the auditorium?
Thank you, Dr. Patrick, for your talk. In one of the slides you have mentioned you use plasma technique to make the thin films, right? My question is, what is the minimum thickness you can get from that technique? And why I'm asking, because we grow silicon and zinc oxide nanowires. So maybe what I was thinking is we can coat them in future end, because it can improve the stability of nanowires in mediums. That's my question. Thank you.
Yeah, no, so there are different ways of plasma, right? So you can do plasma deposition. That is not what we do here. We make use of oxygen plasma actually to etch. So we do a very relatively mild treatment, because if you do a very long treatment, you will actually etch away your surface. So we only apply an oxygen plasma or an air plasma to oxidize the surface to generate it to generate a negative charge. And so we also design positive particles, and then we can add them in one go. And if we want to add then the negatively charged particles, we just need to have a layer of positively charged polymer in between. But we only use the plasma to activate. But indeed, we can do it in principle with any material. So if you would have the nanowires and we activate them with plasma, then you can quite easily coat them with dip coating or spray coating. That should not be a big problem.
Okay, well, let's thank Dr. Patrick Bakrai. Thank you. Just now we will go to the coffee break. So let's be back in 15 minutes. So we're a bit late. So we will be back at 10:55. Thank you. 10:55.
You are listening to the sound of a completely new...
Okay, well, let's start with the second part of the symposium. I will introduce the nice speaker that we have with us. So it's also a very nice example of this combination of company working in scientific, but in a company, and then also sharing or doing also work in the academy. In this kind of topics is very important. So I'm very happy to introduce Dr. Brandon Peterson. Dr. Brandon Peterson did his Bachelor Science Chemistry in Xavier University at Cincinnati, Ohio, in United States. From 2001 to 2009, he was contracted as microbiologist at Procter & Gamble in Cincinnati, Ohio. At 2013, he earned a PhD in medical sciences at the University of Groningen in the Netherlands. And 2016, he was postdoc at the Department of Biomedical Engineering, University of Groningen as well. And in 2018, since 2018, he is faculty member of the University of Groningen. His research aims are about bacterial microbiology and bacterial biofilms. Dr. Brandon is a program leader of the MAN Biomaterials and Microbes program within the Kolf Institute, which facilitates translational application of biomaterials into patients. Brandon Group focuses on bacterial attachment mechanisms to biomaterials and how bacteria adapt to their surroundings as a means for survival. Learning how bacteria adapt to their surroundings is key to combat bacterial infections of biomaterials without the need for antibiotics. So please, let's give a warm applause to Dr. Peterson.
Oh, thank you very much for that wonderful introduction. As mentioned, my talk is going to be a little bit different than usual. I'm going to focus on the microbiology. I am a native English speaker, so if by chance I go a little fast, it's okay to wave in the audience and I will slow down. As you can imagine, when you're speaking native Spanish, you can go rather fast as well. So it's okay to wave at me to get me to slow down. So a little bit about the introduction. It was nicely summarized, but I do want to go over it again because it's critical as to how I came up with the bacterial adaptivity. I was a synthetic organic chemist at Xavier University. And you might realize, how does a synthetic organic chemist become a microbiologist? It's a very interesting transition. Well, I got moved to the company in 2001 as an analytical chemist, basically the starting point of all chemistry in industry. My boss really enjoyed my work, but was a PhD in microbiology. So they moved me to the microbiology department, and I told them, as long as it's how chemical systems interact with biology, I will be okay. If you try to get me to look under a microscope and identify bacteria, I will be useless to you. As it turns out, we had a successful relationship within the company. I was part of the team that created the Crest Pro Health or Oral-B Pro Expert brand that you might see still on the markets today. I was on the team from start to finish there. And when in the company, I got recruited in 2009 to the University of Groningen to do my PhD, which I finished in 2013. They kicked me out of the country for 2 years, but then invited me back in 2016 as a postdoc, and then I became a full-time staff member in 2018, still within the microbiology department. So we've talked today a lot about biomaterials and how you try and make biomaterials for your individual systems. And from the clinical perspective, what we can see up here, I think this is the one up, I'm not sure which, the light's up here. But we can see that there's a lot of different biomaterials for a lot of different types of basically organelles or human parts within the system. I actually teach at the University of Groningen, a medical implants course where we were trying to recreate an entire skeleton solely from biomaterials. And pretty much we could do that except for one or two different pieces, but we could physically create a skeleton with mostly biomaterials. So at this point within the system or within the scientific community, we are capable of basically recreating the entire human body with known biomaterials. However, there's always room for improvement. When you think about biocompatibility or attacking or making anti-fouling, I always say it takes two to tango. Most of the nanomedicine, nanotechnology, the physicists, the chemists, they always look at the anti-fouling properties from the perspective of the material. But it does take two to tango. And that's why we need microbiologists to look at it from the bacteria point of view. And that's where my group comes into play. We look at the interesting parts of the bacteria, how they interact with the nanomaterials, and we go through and we make the adjustments as necessary to the chemists and the physicists so that we have a working mechanism as a team. When you're working with anti-fouling properties, you really want to focus on the prevention of bacterial adhesion. I could go into a long talk about all of the different stages. A little bit of that will be in my afternoon talk today. But for the key perspective here, for the shorter talk, you want to try and look at preventing of bacterial adhesion. That is the quickest way to prevent a bacterial infection on whatever material you are trying to introduce into the system. You might wonder how we got to bacterial adaptivity, or what is bacterial adaptivity. And I started with, this is something from my PhD, this particular figure here, looking at biofilm adaptivity, how biofilms react to their surrounding environments. There's a mechanical interaction, how biofilms interact with physical forces that they may see, but also chemical interactions as to how they will actually react to antibiotics. If we look at it from a bacteria point of view, we can look at it from these three similar situations. We have a physical, which is the mechanical point of view, the chemical, antibiotics, but also a phenotype within the biofilm where bacteria individual strains will change their phenotype due to a specific microenvironment, and then they will switch back when the microenvironment is no longer necessary. So how I came up with the bacterial adaptivity line basically starts from this particular research from a postdoc, from my postdoc year in 2016, where we were looking at mechanical sensitive channels of large conductance. At the time, I had no idea what that was. However, basically what you need to know, again here, when a bacteria is in suspension, the cell wall is basically intact, and all of the channels are closed to prevent osmolarity, to keep the osmotic pressure within the system active. When the bacteria is sent as a surface, there seems to be some type of cellular deformation, cell wall deformation, and that actually opens up this channel of mechanical sensitive of large conductance, and then allows a lot of outflow from the inside of the bacteria to the surrounding environment. So you can see here quite simply that there are two different phenotypes already seen if the bacteria is in suspension versus if a bacteria is actually attached to your surface. Furthermore, we tested this against antibiotics. So we used a system known as IVIS, where we were able to see very close interactions to the surface via gold reflectance. And we were able to see the different types of antibiotic increases as the bacteria got deformed. And we actually used a mutated strain where we actually eliminated the MSCL channel. My poor postdoc was using a bacterial strain from several years ago, and was struggling to figure out why her experiments were not working. She was doing the same procedure over, and she was getting similar results from the parent strain and the mutant strain. Well, when I got here as a postdoc, I asked her when last time she confirmed that she actually still had the mutant strain, and that had not been done. So we verified that the mutant strain had actually reverted back to the parent strain. So we ended up getting the mutant strain again, and then we got these nice results. And she successfully defended her thesis. We've also heard a lot of speakers talk about photothermal particles, nanoparticles. So I thought I would throw this in. This originally wasn't part of my talk, but as we have talked about this, I thought I would also join the party of nanoparticles. So yes, I do have polydopamine particles. We do a thermographs curves, and you can see that, wrong button. We have the temperature, again, looking somewhere between 45, 50 degrees, as Ali had mentioned, but also that you can cycle it on and off, and you do not actually lose the power of these particular dopamine particles. We, of course, looked at the optimum concentration to see what we could do with killing effects, and tested it against what we consider known as the escape panel of bacteria. And these are the bacteria that are highly mutating and highly antimicrobial resistant. So if you can have effects on these particular bacteria that mutate quickly, then you have a higher chance of being effective within the bacterial community. And for the most part, we had a very nice increase, again, with planktonic bacteria in this particular case. One interesting part of this was our particles seemed to enjoy Staph aureus. So we actually got more Staph aureus than we actually put into the system after we put the nanoparticles on there. So that is something that we have to be careful of when we want to try and use the system. But again, my talk is about biofilms. So here we have a confocal image of where we used our polydopamine particles within the presence and absence of particles. And as you can see, there is absolutely no killing in these pictures. For the rookie, you would be very discouraged. For the experienced scientist, we said, why don't we put the nanoparticles in the biofilm and then do it? And now we have complete killing of our system. So we didn't abandon the nanoparticle system. We put the nanoparticles in there, heated it up, and we were able to kill the entire biofilm. And then we thought, well, maybe we did a little bit overpowering of the system. So we only treated it one day with the particles, and the second day we didn't treat it with the particles. And we still got a majority of killing in this particular case. So what we found out was that it's important for this particular system to be inside of the biofilm in order to be effective. It's not going to be effective if it's used as a treatment of a pre-existing biofilm unless you can get the particles inside the biofilm. We also had some talks about why we have particular temperatures and cellular damage. So here we actually did a window of killing, both for fibroblast cells and bacteria. We have the thermographs of the temperature, and we have the coverage and the different volumes of what we have. We were looking for greater than 90% kill of bacteria, which is a 1 log reduction, and a very low acceptance rate of 40% cell coverage, of which we were hoping that we would then get recovery afterwards. So we were looking at an initial drop of, at best, or at worst, 40% coverage, but the recovery afterwards would be higher. And what we found was that with a very small amount of liquid, we were able to get a window where we were able to kill the necessary amount of bacteria, but also maintain a certain amount of cell health that would then be able to recover and regenerate. However, if we had too much of a liquid system, these particular windows would not overlap, and we would not be able to specifically target the bacteria while not harming the cells. So that covers the particle end of things. But now working in the laboratory, this was actually my first project as a PhD student. Not very well received within the department, I must say. But it is still highly cited as far as one of my best publications. And basically what this is from a physical point of view is measuring how much compaction you get when you actually harvest your cells in a centrifuge tube. And you'll notice that I have different geometries. We have a cone tube that we pretty much are very familiar with. We also have a round bottom tube and a spherical tube. And all of those have specific geometries. All of those will create very specific different types of pellets when you try and centrifuge things down. So we did a lot of mathematics and trying to figure out exactly how compact the bacteria were within the pellet. And basically the compaction of the pellet was the results of the experiment. There was no other experiment except trying to figure out how much compaction there was. How we came up with that, we had a reference paper to the particle packing or perfect particle packing, which used nice sines and cosines, but I simplified it to a nice 0.63 for biologists. If you give biologists lots of mathematical systems, they will just throw it out the window. So basically, I simplified all the complex mathematics for physics and gave it a nice 0.63. Then I have the simple volume of the bacterium, basically how big is the bacterium that you're trying to centrifuge down, the number of bacteria in the system, which we counted, and the volume of the pellet, which we measured with a nice little glass blowing technique that I used from chemistry. And basically, the number of bacteria that was in the pellet was relatively stable, no matter what speed we used. However, the volume of the pellet decreased depending on speed, but more importantly, the shape of the container. So the shape of the container would vastly increase the actual compaction of the pellet. And then you can see the result. If you keep one of the number of bacteria the same, the volume of the bacteria was also the same because it was the same bacterium. And you change only the pellet, well, then you increase the compaction. And you'll notice that both this one here and this one here are relatively the same, but they are significantly different, as is the speeds here. So just typing a number into your centrifuge is not enough to actually control your system. Words of warning. Now, why does this matter? Well, we basically bacterial experiments typically have a nice raw standard deviation acceptance rate of about 30%. Now, when you're doing animal studies and you have a standard deviation of 30%, that drastically increases the number of animals you need in the system. And ethical committees do not like that. So the idea is to try and reduce the standard deviation in order to lower the number of animals that you might need in your particular studies and come up with better techniques. And you get the same results with fewer experiments. And if you control the compaction, not necessarily directly, but within a certain range, you end up with very, very small standard deviations. Obviously, these are not 30% of the actual value. And you can also predict basically how well your bacteria is going to mimic the natural environment. And our standard deviations were much, much lower than 30%. They were closer to 5%. But then the question becomes, if you can impact the initial bacteria by centrifugation and its environments, how does it affect the biofilm? So this is where some of your nice physicists and chemists would come in. I did see a nice tour of a compression system. We used a nice low-load compression system with our nice Maxwell elements. And we were actually able to compress biofilms. So I know you're working with harder materials here and have some soft liposomes, but we were actually compressing biofilms and modeling it with 3 elements for simplicity purposes: water, BPS, and cells. So we had 3 elements for those that we continued to label slow, intermediate, and fast. And we looked at, ideally, when my supervisors came up with this idea, they wanted deformation, strange use, sedimentation, and shear rate. Well, after publication of my first one, I was very curious about the centrifugation factor, so I threw that in as an extra. And as it turns out, it was the most important factor in my results. So it didn't even matter what strain I used or the shear rate that I was using in a parallel plate flow chamber. The most important factor of the biofilm formation or the biofilm mechanical properties was actually the compaction factor of the bacteria during the harvesting stage. So then we ended up trying to look at where it's impacting in the biofilm. So in this particular one, we changed the bacterial species. I'm not going to try and have you guys recognize this because I know that you're particles, but these are oral bacteria. So we deformed it with 10% deformation, 20% deformation, and 50% deformation. And as you can see, most of the significant results are in the deeper area here. So within the depths of the biofilm, that's where you're going to see the changes of your actual organization of species within the biofilm. However, when we looked at the centrifugation compaction, and we only had it above and below a certain number, which was halfway between all of the measurements that we had, so that we had equal numbers on both sides for statistics. That's the only reason why we chose that number. We can actually see that at 50%, there is no significant difference whatsoever. But at the top 10% and the top 20%, we do see significant differences as to the relaxation properties of the biofilm. But the question became, the effects of centrifugation were only seen by the harvested bacteria, not the ones that were growing on top of it. And if only the initial bacteria are seen in the centrifuge, how can only the top 20% actually have the mechanical properties? So that gave us a little bit of a confusion for a little while until we thought about the aggregation. Now, when you're centrifuging and creating tight pellets, you're basically ripping off everything on the outer side of your individual colloids, if you think of bacteria as colloids. If you have nanoparticles where you make nice functionality groups on the side that stick out, and then centrifuge to try and get individual types of sizes, you will also rip off your functional groups of your nanoparticles. So it's highly recommended that you do the sizes first and then do the functionality, because if you do it the other way around, you may actually just rip off everything that you spent months trying to optimize. But what we saw here was that, with this particular graph, is that as the compaction number was low, or we had low compaction, the aggregation happened faster within the system, meaning that all of the external components on the cell were able to see all of the other components, create the clusters, and fall out of suspension. When it was a higher compaction rate, it happened longer, and if it was very tight, basically it was outside of our 45-minute window, and there was no aggregation to be seen, meaning that the bacteria ended up to be bald. We took this one step further in my PhD and then identified the different types of materials within the biofilm that would be related to the system. We had the relaxation time components. We did a nice statistical analysis called a principal component analysis, but we found that eDNA was the only system within the matrix that had its own isolated component. And basically from that, when I left after my PhD, the members of the department tried to reproduce my results, but they used these results in a liquid phase, whereas my experiments were performed on agar plates. So as we've been talking about water absorption into systems, you can imagine what happens when water absorbs into a bacteria and how it affects your relaxation times. The times that I had here on agar plates are not consistent with those that were made in a liquid agar system or a liquid parallel plate flow system. Those times would be much, much less because there would be more water introduced into the system. So you do have to be careful. These numbers are published, they are accurate, but they are for a dry agar system. They are not going to be universal for any of the systems. You have to kind of test them to yourself. So this is the question that I'm going to then propose. Could extracellular polymers outside of the bacterium affect bacterial adhesion? And at this point, I would kindly ask not to have pictures taken because I still would like to publish these results. And if by chance these pictures come out, then I am not going to be able to do so. However, I thought you guys would be interested in this, so I put it in anyways. If I can't publish, you're going to have to answer to Helder. So basically, I want to try and go over some of the key methodologies of this particular experiment. Basically, we're making movies of dancing bacteria. And basically, we had a bacteria culturing and harvesting. We exposed them to a treatment, put them under a parallel plate flow chamber under different amounts of stress, had the flow arrested, and then captured their movement on the surface. And then at 50 frames per second, and then kind of watch them dance on the surface as they tried to make themselves more irreversibly attached to the surface. We used two treatments. The first one is DNase. Obviously, as I showed you before, with DNA being a primary component of the extracellular matrix in biofilms, it's also present on the exterior surface of bacteria as an individual. So by cutting the cleavage of the DNA, we could kind of break up the polymers that would be used for bacterial adhesion, and also N-acetylcysteine, or NAC, for the cleavage of disulfide bonds. Both of these are actually used in the clinic for cystic fibrosis patients to break up the mucus within the lungs. So these are well-known drugs that are being used to break up biofilms and matrix materials. So I use these as primary components as examples within their combination as well. And here I have 4 TEM images, or transmission electron micrographs. The top 2 are without N-acetylcysteine. The bottom 2 are with N-acetylcysteine. And you can see that the dye has permeated in the bottom 2. The membrane has been perforated, and both of them are deceased. And we did the analysis of these particular colloid bacterial surfaces. The charges were roughly the same. The diameters were roughly the same. Although the diameters were same, the little haze that's outside of the bacteria is not the same. So although the total size is roughly the same, we did in our modeling system did change the tether length according to what we saw in the TEM. So although these are not statistically different, we did measure out the tether length. But as you can see, if you use N-acetylcysteine or the combination, you can basically damage most of the membrane. We also measured how much sugar was removed for the disulfide bonds, and the eDNA that was removed for the DNase. And here we have some pictures of bacteria dancing, or the maps of their dancing patterns. And these are representative images. We did over 90 bacteria per treatment. So I wasn't going to show you graphs of all 90, but these are what they are. And I know the orange doesn't show up very well in presentations, but I don't have the ability to go ahead and edit to make it darker as there are PDF pictures that are then printed out over and over and over again. But basically what I'm looking at is the area here of total movement, the area here of total movement. And then once you get to N-acetylcysteine, basically what I'm looking for you to see is that the area collapses. There's really no movement with the N-acetylcysteine. However, when you put the DNase on there with the NAC treatment, the movement kind of gets back. And that's under static. And then under maximum shear stress that we've added, which was 50 inverse seconds. Again, as you can imagine, as you increase the stress, as you try and get bacteria to attach to a surface, they're going to attach more tightly, which means they're not going to move as much. So these are always smaller than these. That's the takeaway from here. We did plot the mean square displacement to see how the bacteria was moving in each particular case. And although the total movement, as you saw in the graphs, was relatively different, basically all of the units are roughly the same. So the total mean square displacement is roughly the same from all of the treatments, although the movement patterns were a little bit different and more spread out. So that was an interesting find for us. And then we went to an in silico model, where we ended up modeling the bacteria's attachment to the surface and finding out how many contact points the bacteria actually had to the surface. So basically, our model isn't perfect, but we have concentric rings of 3, 12, 27, and 47 tethers. I didn't bother to put the 47 tethers in the outer ring with all the X's, but you can get the general idea of increasing number of X's around, making it a symmetrical area. And basically, how to maintain the tether length, angle, and force was all part of this particular model. And these were the parameters within the model. And the experimentally determined ones, we have the spring constant, which was taken from the MSD. We have the tether rupture distance, which was variable per treatment, and that was based on the size of the TEM images. And the number of springs attached, we started at 27, but we changed that within the process to make sure that we had an optimized system. So basically what we were looking for, you're looking at a rupture distance, and I have kind of an animation here. It's very rudimentary, but basically after a bacteria reaches that particular rupture distance, it's then going to detach itself from the system. Here, if you change the rupture distance and its length, even though the original one will detach, the other one that has a higher rupture distance will stay on the surface. And then you can also change the initial length, which is what we did, and then you change the breaking point as well. You'll see that. It looks like that was the same slide as the other one, so I didn't necessarily get the right one copied over this morning. But here we have a table of all of our in silico measurements. So again, we mentioned that 27 was the critical one in the table as far as the tethers, but we did test 3, 12, 27, and 47, and then also the rupture strength we had. And basically, from the in silico model, we were trying to match the experimental details that we had. And from that perspective, we were able to then identify the rupture distance length and the number of tethers that were in the system for each of the individual treatments. So here's the no treatment. We had it for the DNA treatment here. And you can see we went from 28 to 32 already. We add NAC, we get it to 35, and the combination we get kind of in between. We didn't have a good fit in this particular case. And even if we went 36, 37, 38, 39, and 40, we ended up with the same stuff. So for simplicity purposes, when we publish, we're going to keep it at 35 and 40 and take the average. But the numbers really didn't change to kind of match the experimental setup of the double. So basically this table here is a summary of that. The only other thing that we added was that the number of bound tethers at the end of the experiment. So we had the rupture distance in silico, and we were able to then see exactly how many tethers were still remaining at the end of the 40 seconds that they were attached to the surface. And basically, for those that were not treated with N-acetylcysteine, although it's very small, you can see that the numbers decreased. And for the N-acetylcysteine, the numbers increased, which would indicate kind of a bond strengthening within the surface. Now, and this is true even though those bacteria are completely dead. So again, how does this come to bacterial adaptivity? Well, most of this is talked basically from the physical chemical standpoint. And if you did this experiment with colloids with the same type of accessibility, you would get roughly the same results. As you end up with shear, the bacteria then adapts the strength and strengthens the bond and shortens the amount of time that it moves. So the bond should trend to require more adhesion force to withstand the more shear. And it's the potential to get closer to the surface as you end up getting the movement of the tethers to go down and get more attached. So this is where I'm going to bring you to the viscoelastic failure point, which I'm hoping most of you are familiar with as chemists and physicists. But bacteria behave as colloids surrounded by the extracellular polymers, which do make them viscoelastic in nature. And basically, we're interested at not necessarily temperature, but in our case, stress. And what we were looking for was the transition between the natural state and how much shear was needed to apply in order to break those springs of viscoelasticity on the surface. When you see the untreated, you do not see a failure point. It doesn't fit the model. It basically can adapt all the way up to 50 inverse seconds. There is a reduction, but there is no failure point. So this is how bacteria can adapt to their surfaces. Once you start manipulating the polymers around them, they no longer have as big of an ability to adapt to their surfaces. If you see the DNase treatment, you see a very large jump in the spring constant here. The NAC treatment is a smaller one. The combined, we don't see any difference in the spring constant, but we do see a difference in the diffusion coefficient. So again, there are ways that the bacteria can adapt. So we don't see anything in the diffusion coefficient with the untreated and the DNase. This one does have something that is listed, although it's very small. And you can see that in the spring constant, you have very, very large indications of the transition point between where the viscoelastic failure point would be. So revisiting this particular graph again, we have the treatments where we have the DNase and the DNase that have the diffusion coefficient, or the spring constant be adapted in very, very large ways. But then we have the untreated and the NAC treatment where the diffusion coefficient was not actually talking about. And looking at these, they don't seem to match up, except under the high stress, you do see these to be a little bit larger, but under the static adhesion, the red arrows don't really seem to be telling you much. So note the gray scatter of the untreated and the NAC exceed the scatter of both the DNase and the DNase treatments. So again, using the NAC treatment, which does make them dead, by the way, allows for the excessive scatter rather than having the DNase and the DNase plus, which they're still alive. And then you can then see that they're adapting to the surface. So with that, I would like to have my acknowledgements. As I've been mostly within the university, I've had most university funding for the most part. I've had a couple of Erasmus funding students as well, so I do have to add that to the slides for that type of purpose, but I want to acknowledge the Erasmus Plus there and the CSC scholarships. But thank you for the attention. I have my LinkedIn research and Twitter, and now I'm open for questions.
Hi, thank you. I have only one question. How do you or did you calculate the stress?
So the stress, we use a parallel plate flow chamber. So we have known dimensions, and we calculate via a pump where we have a calibrated system. Every time we use the system, we calculate how many mils per minute, and then that gets calculated down to the shear stress that we are interested in.
Okay, nice. That's a really nice way to calculate. Yeah, thank you.
It also makes it more consistent between experiments if the tubing is not precise in the pump.
Yeah, exactly. Yeah. Thank you.
More questions?
Thank you very much for your presentation. I'm wondering if these results you show, have you tried to, this is only with one single species, bacterial species?
Yeah.
And did you try using different bacteria?
Yeah,
and
not
also...
in this particular one, but I do have a current PhD student doing this for the entire ESCAPE panel, which also does both spheres and rod-shaped bacteria. Now, our program is really designed for center of mass. So when a rod rotates, I'm expecting there to be some issues with the data, but we're giving it a try anyways. But we do have multiple species of coccoid or sphere bacteria that we are trying with this PhD student.
Okay, because I'm wondering, because in the oral cavity we have a lot of bacteria.
Yes.
The biofilm is very complex, a lot of hundred of different
species. Yes, over 1,000 species.
Yes. So I wonder if you try to see something. Okay.
Right now we're with the ESCAPE panel, but you know, if things work out, we might throw in a couple oral bacteria as well.
Okay, thank you you very much.
Thank you, Dr. Peterson, for your talk.
Yeah.
My question is, for example, when we work with different semiconductor materials, the interaction we have to increase the interaction, it should be faster, for example. So how we can make the interaction of these bacteria, different kind of bacteria, as faster as possible, because it will enhance the properties of biosensing. And the second thing is, do we need smoother surface or the rougher surface for these kind of interactions? That's all. Thank you.
Yes. So you will enjoy my talk in the evening session if you're there. I have a whole section on surface modifications that I use in the course. There is no easy answer with that because different bacteria react differently. So it depends on your particular species, whether or not you want a rough surface or a smooth surface. Generally speaking, a smooth surface will be better. As far as your other comment about, can you just, the topic again? Yeah, so increase, the different materials, yes. So basically what we're interested in now is looking at the number of attachments and how strong those attachments are. So we're not, right now we're under a microscope with glass, which is not an optimal system for biomaterials. But our idea is, from the fundamental point of view, understand these particular bonds and then translate them to other dipole moments when we can measure them with an understanding. If we go in blind and switch materials, it's going to be a lot harder for us to find a connection. But I want to say that usually you don't want to have a strong connection because you'll either leave dead bacteria residual on your surface. So if you you spend a lot of time making a nice coating, you kill the bacteria settles and has a strong adhesion. The next bacteria is not going to see your coating, it's going to see the dead bacteria, which is then going to attach.
Thank you very much, Brandon, for a very nice talk. So good that you are doing, looking at the bacterial bond with a surface, let's say. Mm-hmm. I don't think there are a lot of requirements with respect to the surface. It doesn't have to be transparent. You're just using fluorescence, right?
No, these are actually transparent at the moment. So we're using light microscopy at the moment. We do have a system that we can do it one at higher speeds in the system, and we can adapt it for fluorescence if necessary, so that we can still grab these particular bacteria for different types of surfaces. Okay. But right now, it's currently only light.
Okay, but it will be nice to move to fluorescence because
absolutely.
I was wondering if you can do this. Can you now visualize two bacteria, neighboring bacteria in a biofilm, and try and predict what kind of interactions do they have?
Well, if you're going inside of a biofilm, now you're going to need a confocal of you're going to have to go into a confocal type of microscopy. And then you're going to have limits of frames per second within the confocal. I would prefer to keep it outside of the biofilm for now for speed purposes, but you could look at interactions between neighboring bacteria. So these bacteria are isolated from each other so that there's no impact of the neighboring bacteria, but there is no real issue of trying to grab neighboring bacteria and looking at the differences between those to see if there is an impact of neighboring bacteria on the surface, other than the limitations of MATLAB.
All right, thank you very much. Yeah.
He was also my PhD supervisor, so he's very familiar with this work. Yes.
Thanks, Brandon.
Yes. Thank you very much.
Hi, good morning, everyone again. So this is the time in which Sartak Misra is going to present another exciting talk. So I'm going to introduce Professor Sartak. He joined the University of Twente in 2009, and he's currently a full professor in the Department of Biomechanical Engineering within the Faculty of Engineering Technology. He is also affiliated with the Department of Biomedical Engineering at the University of Groningen and University Medical Center of Groningen. He is leading the Surgical Robotics Laboratory. Sartak obtained his doctoral degree in the Department of Mechanical Engineering at the Johns Hopkins University in Baltimore, USA. Prior to commencing his studies at Johns Hopkins, he worked for 3 years as a dynamics and controls analyst in McDonnell Trailer and Associates on the International Space Station program. Sartak received his master's degree in engineering in mechanical engineering from McGill University in Montreal, Canada. He has been the recipient of multiple awards, prestigious awards in the Netherlands, Europe, and also in Canada and the United States, including the NASA Space Flight Awareness Award. Sartak's broad research interests are primarily in the area of applied mechanics at both macro and micro scales. He is interested in the modeling and control of electromechanical systems with applications to medical robotics. Thank you, Professor Sartax. The microphone is all yours.
Well, thank you for the very kind introduction, and also thank you to the organizers for hosting us and being so hospitable. It's been a wonderful experience. So this talk is going to be probably very different from what all of you have heard till now. And but what I really want that you take away from all of this is that the techniques, methods that I'll walk you through, hopefully that will inspire you or also get, you can use some of those things in your work. So the reason why this talk is going to be different is because I'm an engineer by training, and I was introduced very kindly, and I did not expect that they would talk about my background. But that is why I thought that in order to set up the stage for what is going to happen in this presentation, so I thought just to give you where do I come from and where is this all going. So I studied in Canada, and I studied at McGill, and my research and thesis there looked at the dynamics of multi-body space systems. And what this is basically large space structures, and that's the Cassini spacecraft there. So I looked at how the attitude dynamics of these structures when they're orbiting around the Earth, how do you model that? How do you understand that? After that, I decided to go and work in industry. And I worked for about 3.5 years on the International Space Station program. And the reason why I worked there is because Canada actually owns and operates all the space robots on the space station. So, and I spent some time there as a robotics analyst, both in Montreal and in Houston. After that, I decided to go back to school again and pursue a doctoral degree, and this time in Baltimore at Johns Hopkins. And my thesis there looked at the interactions of surgical tools, soft tissues. So from space robotics, I moved into medical robotics. And this was basically developing planners to train clinicians. And as it was when I was introduced, after this, I decided to move to the Netherlands, and I joined the University of Twente, and a few years later also the University Medical Center Groningen. So in a few words, if I were to describe what goes on in the lab, is basically developing new ways, novel techniques to reach challenging locations within the body. We have lab locations both at the University of Twente and also at the University Medical Center Groningen. So the way we think about doing these things is first by developing preoperative plans. So we live right now, and we're in Mexico City in a hotel. And if I were to figure out how to get from my hotel to this place, I would put it on Google Maps, and it would figure out a plan from A to B. And that's kind of exactly what we do. Let's say a clinician wants to reach a certain point in the body. How do you make this plan to go from point A to point B? You have to account for what tissues are in between, how is your instrument going to deflect. So you use various non-imaging modalities to come up with these plans. You use MR, CT, ultrasound, use these models, methods to develop these computational models, and then use these plans and models to control a variety of instruments. So these could be flexible devices, needles, snake-like robots, bio-inspired structures like the little kind of thing that's moving there in the corner there. These can be actuated using various kinds of techniques, magnetic, acoustics. And then we go down also several orders of magnitude where we're trying to design, fabricate, and steer these micro-robotic systems. So what this talk focuses on is primarily how we design these things and how we control these things. And the idea is that these devices would one day enter into our body. So how does this, what is the thought process of that? So you have a flexible device that is inserted into the body and kind of delivers a drug somewhere deep inside the body. And then you apply some actuation method, and this could be magnetic fields, acoustic fields, and these are then used to guide your robot into the deep-seated region in the body. And so what this talk focuses on is primarily these kinds of things where you want to fabricate them, and how do you want to actuate them. I have no idea what's coming up there, but it's not coming on my screen. So, so this talk will focus on magnetic actuation, acoustic actuation, and then once you have this, how do you actually image robots inside the body? So the first one is on magnetically steering these miniaturized agents. So magnetic steering can happen in many ways. You can apply magnetic fields to kind of actuate them. And these are few classes of these kinds of robots. So this is an example of a hydrogel gripper, a paramagnetic particle or a contrast agent. This is something we fabricate in the clean room, a bio-inspired structure called magnetosperm. So the idea is that these robots have magnetic material on them. You turn on your device, and these robots can then be steered in a certain direction, but also actuated in a certain way. Alternatively, you could have self-propelled structures, meaning that they take energy from the environment and then start moving. And so two examples of these are a microjet, which actually is in the Guinness Book of World Records as the world's smallest jet engine. And I'll get to that, how this thing moves, or these bacteria. So we cultivate also bacteria with magnetic material on them. So these are self-propelled structures, but then you apply magnetic fields to kind of point them in a certain direction, like a compass, and then they can move as you want them to move. So the next, I think 3 or 4 slides is probably the only, being an engineer, the only slides that have some 1 or 2 equations. So please bear with me, but I just really wanted to kind of show you how we think about doing this, how we think about controlling these systems. So in order to control a robot inside the body or anywhere else, you need to figure out first how to track them. So you need to know where it is. So you can do that with camera images, microscope, or in this case, an ultrasound image. So once you know where this robot is, you need to control it. So let's say you say you wanted to go from A to B, one point to another point. You need to then give what your point B is. So what is the reference position and what is the position where you are in? So you have to know where you are. You want to know where you go, and you apply, let's say in this way, a control law. So this is called a PID control law. Maybe some of you in the audience are engineers here, and this is in the most simplest form how you control a micro robotic system. This controller spits out certain forces. What you do next is do a force to current mapping. So these forces are something that you have in your software, and then you have to convert this into currents, because the currents are the one that drive the coils. And these currents are then fed to the electromagnetic coils, which can then actually move your robot, let's say, along this path. So this is the basic framework of how you can image and control a micro robotic system. You can get a bit more fancier. Then now let's say, when you insert something into the body, there are uncertainties. You don't know what's going to happen. So this is a real experiment. This is a microparticle, and the task given is to go from one point here to this green circle. There are uncertainties. This is unplanned, what's happening around it. So it has to completely replan itself and then reach the target location. So that's one thing you need to develop is motion planners. You can also do multi-agent control. Let's say you have several agents and you want to move them around. So in this case, there are 2 agents, and you want the agent on the top to move in a big square and the agent in this center to move in a center square. While here you want to independently control them. So here in this case, we wanted to write UT. So how do you, there are ways in which you can also develop these kinds of systems. So with this framework in mind, what I'd like to do next is kind of walk you through a couple of examples of these micro agent systems. So the first one are these micro grippers. So these are PNIPAM grippers, which Patrick introduced. So basically, but what has changed in this is that we've put magnetic material in it. And it has this stiff polymer in there that basically then acts like a finger. So you have PNIPAM, which is the hydrogel, which responds to heat. And then you have these little phalanges here, which kind of can close. So when you increase the temperature, they close. When you cool them up, they open. And you can keep doing this on and off. Okay? And it's doped with magnetic material. So the reason why we put magnetic material in there is so that we could control it. So in the first example here, so using the basic framework that I showed you, the video on the top basically says, okay, we don't apply any external disturbance. Can we move it around in a circle? And In the bottom example here, the student was using a pencil and kind of disturbing the Petri dish. And the idea was to show that in spite of disturbances around, you could still move this this hydrogel, this gripper in a kind of a controlled manner. So this was to show motion control. Next, we got a little bit fancier. So what's happening here, the task given to the micro gripper was take the pink circle, put it in the pink ball, put it in the pink circle, the blue ball in the blue circle, and the green ball in the green circle. And this is all happening now autonomously. So there's no human in the loop here. So with cameras in there and this tracking it, and then we increase the temperature, it opens up, and then we cool it, it opens up, and then drops the bead, and then restarts this procedure. And the idea was to show that a device like this can be used for potentially for biopsies. And it'll continue doing this, and it'll go into take the green circle, green ball, green bead, and then put it in the green circle. So this combines vision control and also motion planning here. Next, we did the same kind of experiments, but with porcine tissue. So this is porcine tissue or pig fat that's in the background, but there's uncertainty. So there are these balls moving, and the task given to this gripper was avoid these balls and reach your target. And once you're at the target there, close yourself up, and after a while, then go back to your next target point. So these target points are kind of randomly given. These balls that are moving are randomly there. It has to figure out how to avoid this. And the reason we want to show this is that, you know, we kind of plan for everything that's going to happen in the body, but when there are uncertainties and disturbances, we can replan ourselves and reach our target location. So the student who was doing this had, I guess, a lot of time on hand. So what he did next is that he decided, I'm going to play Pac-Man with this. And so what he tells here that this is the pack, this is your little gripper, avoid these ghosts and move to a target location. And the idea was to show here that you can embody intelligence into these inanimate grippers. And he keeps playing it. Sometimes he loses, sometimes he wins. So it's a game that's going on. So, and sometimes these ghosts are able to kind of catch on to him. So just to give you an idea of how we use these kinds of micro grippers. So the next step that we're doing on this is trying to miniaturize these further and do also experiments in human cadavers also. The next example I'd like to share with you is this microjet. So this is an example of a self-propelled micro-robot. So what it has is that it has a platinum kind of shell on the top, and it has iron to make it magnetic. And the reason why we have platinum there is that when platinum reacts with hydrogen peroxide, it ejects bubbles. And there is some amount of hydrogen peroxide also in contact lens fluid. So the idea was to show that you can move these kinds of jets in a medium. One of the steps in all of this is that we need to calculate what the dipole moment is, which is this m value. So maybe physicists here might know this term. And we do this in an indirect manner where we move this jet on a curved path and change the direction of field. And based on this this d value, you can plug it in this equation here and find this m value. And this m value is a key component when you want to do control. So what you see in this video next is that we're tracking this jet moving in a hydrogen peroxide sample, and the task given to it is move to this small green, blue circle. We also did experiments where you, change the flow rate. So there's flow moving in through the into this little channel here. So there's flow rate going in one direction. There's flow rate, and we show that when there's flow rate going on, we're able to track this microjet and move it in a controlled manner. And even when it's opposing flow rate, we can also move this microjet. What next we wanted to do is show that we can do human in the loop control. And what you see here is basically this microjet moving through this little maze, while a human here is controlling it with this joystick. And the task given to the human is get as closely as fast to the end of this maze without touching any of the walls. And the objective here was to show that you can move these kinds of microjets with autonomously or with also human in the loop. The next example I'd like to share with you is this magnetosperm. So this is something we developed in our clean room. Is basically has a kind of a cobalt nickel head, which makes it magnetic, and then this kind of flagellar long thin tail. It is 6 times larger than a human sperm cell, but we thought we got inspiration from the human sperm cell. And it has this photoresist body and tail, which kind of flaps. So when you apply oscillating fields, this flapping tail moves and it propels itself forward. What the plot here on the bottom right is showing is that when you increase the frequency of the oscillating field, the robot moves faster, magnetosperm moves faster, but the magnetic field remains constant. What that means is that propulsion is happening because of the tail moving, not because of a magnetic field gradient that's pulling it. And the way we do this is basically you apply oscillating field to 2 of these to 2 of the coils, and you apply a constant field to the other 2 coils, to kind of point it in a certain direction, then start oscillating the other 2 coils. That moves the tail, and you move forward. If you want to change direction, you change direction of the coil, of the current in the coils, and that's what you see in this video here in the bottom, where the magnetosperm moves to different points within your workspace. So what this slide shows is basically kind of compares all the different micro robots that we had worked on a few years ago. So what the key thing to take away from all of this is that the magnetotactic bacteria, so the things that are available in nature, are the most efficient ones, so that they move at the quickest body lengths per second. That's 1.7. So if I were to increase the concentration of hydrogen peroxide, my microjet would move faster. If I were to have larger field gradients, the cluster of nanoparticles would move faster. And the same works for also the Janus micromotor, which is essentially one end of it is coated with platinum. In order to do this, over the years, we have developed lots of different magnetic actuation systems, and this just shows us some highlight or one slide in how these magnetic systems look like, depending on the application that you have in mind. If you want to work with a Petri dish, you have the one on the top left. So if you need really high field or field gradients, use the one, this BATMAX system. So just the different kinds of magnetic actuation systems that we have in the lab. So these are kind of external magnetic actuation systems, things that are outside the sample space. But what if we could have magnetic actuation systems inside inside the body or close to the workspace? And that's this kind of a system. So basically, you take a kind of flexible device and attach magnetic coils at the end of it. And then you can insert it into the body. And then when you open up the magnetic fields, they kind of open up like a flower. And then you can deploy your microagent and then control them. That's what this next one shows here, where basically we're able to control a single microparticle, moving it in a kind of a triangular fashion. This is a proof of concept, but basically the idea is to show that you can take magnetic coil systems also inside your body and move micro robots. And systems like these, I will discuss more in the lecture tomorrow about these various flexible devices. We also develop different kinds of micro robots that can move in one direction or another direction. So this is an example of basically a silicon-based little sphere, which has a kind of a kink or a sphere at the end of it, and we apply rotating fields. And what we notice is that you can get this kind of like a corkscrew motion with this kind of a robot. But what is even more interesting with this is that if you change the angle of the kink at the top, you can get bidirectional motion. So by having this, the first one, the type 1 is kind of at around, I think, 30 degrees, and the one on type 2 is 45 degrees. And when you apply the rotating fields, but also change the precision field, so not just rotation, but the angle at which this rotation is happening, you can get bidirectional motion. So this is something that, so basically you're showing independent control of two robots without really changing much of the material properties and with the same magnetic field. And that's what's interesting about these kinds of systems. We also develop bio-inspired structures. So this is an example of using basically EcoFlex or silicon-based structures, put magnetic material in them, and then magnetize them in a very specific direction in order to get bio-inspired structures. And we take inspiration from things like millipedes or snails or inchworms to develop these kinds of devices. And the next slide kind of shows. The first one on the left is showing a caterpillar-like motion. So you apply magnetic fields, and its tails are kind of moving, while the one on the bottom right is this kind of moves like a turtle. It can do locomotion, so you can tell it go from one point to another point, and then it goes there, has these inchworm-like motions. So basically, magnetic fields are being applied in a certain direction, and then you can ask it to move. So in the bottom two videos, what we're showing is that you have to avoid these blue blobs, so avoid these obstacles, and then move to its designated path. So in addition to locomotion, they can also do certain tasks. So the next video kind of shows that. So in the video on the left side is basically telling, pick up this little rod, avoid these obstacles, and then move to your target location. While the video on the right is saying you have this little bead, capture it with your gripper in the top, and then put it in the target box. And basically the white part is non-magnetic, while the tails or the legs are magnetic. In addition, we also developed these other kinds of snake-like structures. So the video on the left-hand side is basically several of these magnetic elements connected by non-magnetic elements, and it moves like a snake from dry surface to wet surface. Once it gets to wet surface, it also releases certain drugs or a drug stimulant, and then can combine itself together. While the video on the right is basically showing these simple structures that can move on mucoadhesive surfaces, both in two-dimensional and three-dimensional kind of idea. But the idea is the same: silicon-based materials, put magnetic material, magnetize them in very specific directions, and then put them in a magnetic field. So the video now on the right-hand side is basically it grabs onto a slab of a gel capsule, grabs it, moves it, and then squeezes it to release a drug or a drug stimulant. We also develop these origami structures, or these are polyvinyl alcohol-based structures. So when they respond to heat, they open up. When you cool them, they can again close. And the idea here to show that that it can release something it's basically origami inspired, and then can close up again, similar to a gripper, but made from a different material. And we also did tests in the GI tract as a simulant. The one on the middle video is basically showing, middle picture is basically showing a colonos or an endoscope model, while we also did experiments with CT scanner to track this object while it's being moved. Next, what I like to do is, so we've talked about using magnetics as the actuation mechanism for structures, but magnetics is not the only way to actuate structures. You can use sound waves also. So sound waves, when you propagate them through a medium, they can move things. They can move them by acoustic streaming. So waves get formed, and then these waves can move objects, or they are by acoustic radiation forces, meaning that the sound waves penetrate the body or the object that you have, and that causes it to, because it moves at a certain frequency, and then on that frequency, these objects can move. So there are two ways of kind of actuating objects with sound. So the video here basically shows a structure that we fabricate in the clean room, a microfluidic channel with these kinds of wedges on the top. And then we apply sound waves to it. So once you apply sound waves of a certain frequency, you see that these fluorescent polystyrene beads move in one direction. And when you change the frequency of your apply sound waves, they can move in another direction. What you see these vortices happening at those corners are basically the acoustic streaming happening. What you can also do with this is that the video on the bottom right is basically moving a small sperm-like robot through this medium by acoustic streaming. We also develop propellers. So what you see in this example here is basically in the clean room, we develop these propellers that can trap bubbles. So these are basically made on using 3D lithography or direct laser writing or photolithography. So what this is that there's a cavity in these capsules that basically traps the bubbles. And then when you apply frequency sound waves at a certain frequency, these bubbles oscillate and they can move around. And that's what's happening in this video here. So what you can do next is also use this to have these materials and then put magnetic material on them, and then they become magnetoacoustic. So in the first case, the acoustic is off and magnet is off, so it just kind of stays there, floats. Then you turn the acoustics on, so it propels, rotates. Then you turn the magnetics on and you apply magnetic fields, and then you can kind of guide it to a certain place, turn it, rotate it. So the idea was to show that you can use both magnetics and acoustics to steer objects. So when we were doing this work, this was before the pandemic, there was a program on Netflix called Squid going on. So that made Squid Games going on, and that got us inspired to make cephalopod-like robots. So cephalopods have flaps and they kind of flap themselves and move. So what this structure has is basically several of these capsules that can trap bubbles. And then we apply field oscillating acoustic waves that causes these bubbles to move. And that can move this kind of cephalopod inspired robot called Cephalobot. And that's what you see in this video here. Basically, the acoustics, it has a combination of acoustic streaming and acoustic radiation forces. And you can guide them to move in very specific ways. It's writing SRL there, or Surgical Robotics Lab. But basically, the idea is to show that acoustics can also be used as a way to actuate objects. So the first talk this morning was about optical tweezers, but you can also have acoustic tweezers. So these are a set of these acoustic piezoelectric elements. Basically, you can see that you can trap objects in a certain point. You can manipulate them. They can work also in water. But again, the idea here was to show that you can use acoustics as a way to trap objects, manipulate objects, move them inside the body, and the frequency at which this moves is also harmless to the human body. So you have magnetics, you have acoustics as a way to actuate objects. Okay, so that's great. So you can move things, but then you must be able to see them once they're inside the body. So how do we do that? So one of the most common ways to do that would be because of using ultrasound. And the reason why we use ultrasound is because you can get images in real time, so you don't have to wait. There's no damage to the patient, and it's also relatively of low cost. So what you see in these videos here, the video on the left-hand side is showing using ultrasound to track 2D objects, while the video on the right is basically tracking an object in 3D using ultrasound. And this is a micro-sized particle, but we also track microjets. Microjets definitely become much more complicated to move because of the bubbles that come out, as you can see in the videos on the bottom left and right. So ultrasound is great, but it still has its challenges. So what we developed next is this system where a robot holds the ultrasound probe and then moves and then tracks the object. But when I started this talk, I mentioned first you need to know where you're going. So the first thing that we do is make a plan. So in this case, this mannequin or this phantom is here, and we want to track an object in the jugular vein that's here. So first it must make a plan. So how does it do that? There's a 3D camera attached to it. It kind of scans the surface and figures out that this is where I need to go. And then once it takes that plan, so that's your preoperative plan, then it kind of moves. And then what you see on the video or the pane happening in the right is basically it's segmenting, slicing out, this is where I need to go. And once it knows that, it starts its procedure. So that's kind of what it's showing here. And the next part of the video basically shows you how you, now in this case, you're tracking these micro-sized objects, both in phantoms, but also in this is a slab of porcine tissue. You can track one object, one micro-sized agent, several of them many times. But the idea was to show that with ultrasound as an imaging modality, you can track objects inside the body in a reliable manner. So that's what we wanted to show here. So ultrasound images are great, but they are low resolution. They're not easy to see. Once you're deep inside the body, it's not easy to see objects. You can use microscopes, but then you're only on the surface. You get very good resolution. So, but you can't see things inside the body, and that's where you want to track your objects. So what we have decided a few years ago is to look at another technique and develop this. Is called multicolor fluorescence imaging. So there's fluorescence imaging, which you're all are aware of, but you want to track several objects at the same time. So you want to track your robot, you want to track where it is, you want to track your target, so it has to go a certain place, and you want to know what your environment is. You want to know what's in your surrounding. So you want to track all of these things in real time, so that you can move things in real time. So what we have developed is custom-made setup where we develop a method to track objects. In this case, they were custom-made electrospun fibers, but also micro robots that was made to target a specific HeLa cell in a microfluidic channel. And we coat these objects with different fluorophores, so we can track them individually in a round-robin manner, and then process this in real time to track the object. And what we noticed with our system is that there was no crosstalk observed. That means you could track these objects separately, and also you could track both the object and also the environment in this environment, and also the HeLa cells, which is your target to deliver your drugs. So what we're doing now with this project is basically to miniaturize the system, put it on a probe that you can insert into the body. With that, I'd like to come to an end of my presentation. But before I leave, I'd like to share with you a short video that was mentioned briefly to you yesterday, but I'd like to actually show the video to you. So this is a movie that was mentioned last time or yesterday, called The Fantastic Voyage, which was a long time ago. And so the story, just for if you were not there yesterday, was basically the president had a clot that was in his body, and the Russians, being the bad guys always, they had put it in his body, and the scientists had to go to prevent it before it reaches the brain. And I'll show you an excerpt of this video, and maybe you can see when you see some of these robots in there, whatever they show, that maybe they look very similar to some of the things that I showed you in my presentation.
You are listening to the sound of a completely new screen experience, a startling new kind of excitement. As 20th Century Fox plunges you into the most incredible adventure that man could ever achieve. But now, a film called Fantastic Voyage has broken through in an unexpected direction to create an adventure of astonishing suspense and beauty.
One of the miracles of the universe.
Off on a fantastic voyage, actually entering inside the human body, exploring an unknown universe, unknown dangers.
They're tightening. I can't breathe.
So I think those look very close to the grippers that we have been making. But this was 1966, and that's why the effects were, as you can see, what they are. But it is a movie worth watching if you can find it. But with that, I'd like to close, and thank you very much for your attention, sticking around. I know we're above time, but I'd be happy to take any questions you might have. I'm also here during, well, not here in the afternoon, but here tomorrow. And tomorrow I'll be covering some of the other topics that's going on in this slide here. But thank you again very much.
Thank you, Sartak, for this astonishing, impressive talk. You said that this was a very different talk from the others. Yes, it is, but definitely I'm sure it has impressed us. So robots are now with us, and the fantastic voyage is real. And we are not so far from science fictions now. So thank you very much. And please, who wants to ask something? We are so impressed. We have no words. Yes, there's one.
Thank you for the talk. I was just wondering, can you also work with shockwaves? And if so, what could be the difference or the disadvantages and advantages against sound waves and shock waves? And I have another question, which is which is the minimum size you can work with?
Thanks for the question. So the question was, can I work with shockwaves? Sure, I can work with shockwaves, but the key in all of this is it has to be a certain frequency. So we operate in the kilohertz range, so you don't hear that. We do not want to operate in the megahertz range because that's where the imaging happens. So shockwaves are at a much lower frequency. So if you have a much lower frequency, your size of your object has to become bigger. So that is the main thing. So we want to have a certain size, you want to operate at a certain resonant frequency, and you want this to be consistent. And that's the reason why we operate at the kilohertz range. When we were doing these experiments, we can also change the frequency. When you go down to also lower frequency, at certain frequencies, you hear kind of an annoying sound, or you hear a kind of that kind of sound. So these are the various things when you go down to lower frequency happens.
Okay.
Thank you.
And well, the second one.
Yeah, sure.
Yeah. I don't know if you heard, but what is the minimum size you can control?
So the minimum size of the objects we can, we are only limited by the size at which you can image. So with the multifluorescence microscopy, we can go down to track objects that are of 1 micron. And which are inside the body. So with 1 micron objects, you can track them in real time. And also control them. So for our interest is not just imaging them, but also controlling them accurately. So that's where we are right now, and that's, I think, the best you can do in the field of microrobotics. So you can use other imaging modalities like TEM and SEM and all these, go down much smaller, but those are not real time. You can't use them in anything in the body, right? So it's around 1 to 2 microns.
Okay, thank you.
Thank you. There's one here and one there.
Thank you. Excellent presentation. I just wonder, I mean, talking about the fantastic voice that you mentioned and the famous scene where this antibodies attached to Raquel, what you remember that scene, right?
Yes.
So could it be possible as a similar problem with this tiny robots, nanorobots, whatever, I mean, interact with antibodies or other parts of the fluid in blood or in serum, and would that pose a real danger or problem for your results? I mean, that's the question.
Thank you for the question. So I think if I understood you correctly, if I have to break it down, the question basically. So there's fluid flow in the system. The blood moves at a very fast pace. And it's very hard to oppose the blood flow and do something. So if these robots were to get trapped in in our circulatory system, and they might reach in a certain bad place, let's say the brain, that might be not something you want. So what we envision as our system happening, and that's why I had that little animation in the start. Basically, you would have a delivery system that would get you as close as possible where you want to, and then the next few millimeters or a centimeter, you would guide them precisely using magnetic fields. So I don't think we are at the fantastic voyage kind of thing where you can just leave them into the circulatory system and they just move around, because the fluid flow that we have is very fast, and it's hard to overcome it. And so that's why I think the step that is basically is to get as close as you want and then release your drugs, or move your robot. So I didn't have time here to show this, but sorry, but I think some of our robots can grab onto like drugs like doxorubicin. These are anti-cancer drugs, and also release them precisely at various locations. The grippers that we had, basically it was inspired from doing biopsy. So if you have, I hope not, but if you know what, in a biopsy, they basically insert a needle into the body and grab onto tissue. Depending on where in the body it is, it's not fun. So what this does is that basically you could take these robots, you could get them inside the body, either swallow them or get them in, and then they would grab onto tissue and you just take them out. And that's where this idea comes from. So, and you could not just grab onto tissue, but deliver something also. I think there was one here.
Hi, thank you. Maybe I'm the same question but has your research already been tested in humans or animals?
Okay, so in the talk tomorrow, I will show you some of the robots devices that are in preclinical trials. This stuff is quite far away to do any preclinical studies. We are just not there yet. But some of our other devices, the snake-like stuff, the delivery devices are actually in some of them are in preclinical studies. There's a spin-off company from the lab that develops magnetic actuation systems. So that is tested on animal cadavers, small animal studies, like rats, mice, and also in human cadavers. So that's, but this stuff not yet.
Thank you.
You're welcome.
Thank you, Dr. Sarthak, for your talk. Amazing talk, actually. Quite different from all other topics, but I think something new for many people here. I have three curiosities. The number one, for example, you use the mathematical equations, you use the tracking system, then you have the imaging, and then finally you use the system, for example, magnetism to control the flow. Always there is some kind of errors in the system when we have, for example, alignments, and we can have errors. For example, you have shown in one of the slides, you have in micrometers or nanometers. So you can use some kind of feedback loops. Do you use them? And if yes, in how much time intervals you use them?
Okay, so I skipped over the very technical details here, but all of these have a closed loop or a feedback going on. So the feedback is going on because we have imaging there. So the reference position, and there was a tracked position I had somewhere in the slide. So those two are always being compared, and the error between them is being fed back into the control. So there's always closed loop going on. The controller works at a very high frequency. The biggest limitation we have is in the imaging, because imaging happens at, let's say, if ultrasound works at 30 frames per second, cameras work much faster. Fluorescence imaging can also work faster. So 30 frames per second is fine for the applications that we have in mind, because we have these micro robotic systems that don't need to move very fast, but we need them to move precisely. So accuracy more important than speed. Yeah, that was
right. And for multiple fluorescence techniques, for example, what could be the lifetime of the materials you're using? Because fluorescence has a lifetime.
Right.
So what is the minimum lifetime you're using?
So in the examples that I showed, so for the purposes of our experiments, yeah, they last a couple of days even. Some of them last a few hours. So it's for the running the experiments in a reliable manner, they work.
Perfect. And the last one, just out of curiosity, I hope it never happens, but while operating or while having the injection of nanorobots, there is some kind of failure in the nanorobots, and they're inside the body. So what could be the plan to take them out?
So some of them are, so tomorrow I will show you one video of a system where you can use your delivery device as not just a delivery system, but also a retrieval system. So you can inject capsules, but also take them out. We would like to design most of our structures as biocompatible structures or biodegradable structures. So the contrast agents or the microparticles are used in clinical practice, so that's fine. The hydrogel grippers are also fine. You would not want your microjet stuck in your eye or anywhere else, but we have a method where you can eject stuff out also by applying magnetic fields in a certain manner. Thank you.
Any other question? The last one? Here. Just ask, it's here.
Thank you so much, Dr. Sartek. My question is, once these mini robots reach the target and do their job, what happened with them? They can be like eliminated inside the body or recovered?
As I was mentioning to the last gentleman who asked me the question, so with our delivery system is also used as a retrieval system. So the talk tomorrow, I will show an example where the delivery system kind of injects the capsule and also takes it back. Most of our systems are designed to be kind of biodegradable or biocompatible. So most of them are, except as I told the gentleman there, the microjet, or you wouldn't want a bacteria kind of stuck inside somewhere. So that you'd have to wait to reliably get them out. And the way to get them out is basically the same way they got in, the same way you get them out. So I had an example of this Millimax system where the coils open up, and so there's a channel in there that not just delivers the microparticle, but also plucks it away once you moved it around. So that's the escape mechanism or escape route.
Well, let's put our hands together for this amazing talk of Sartak Misra. Thank you very much, and don't miss his lecture for tomorrow. I'm sure it's going to be as exciting as today.
And very different also.
Well, so expect more surprises. Thank you very much. Let's go with the next session. This is also gonna be a different session, so be ready for it.
Hola, hola. Buenas tardes a todos y a todas. Yo les recomendaría que no se vayan, porque ahora vamos a hablar de oportunidades para que vayan a hacer el doctorado, o en este caso el doble doctorado en Groningen. Entonces, bueno, primero que nada, me presento. Soy Luis Andrés González, soy el representante de la Universidad de Groningen en México. Y básicamente dentro de mis actividades, que son varias, una de ellas es la de promocionar y ayudar administrativamente a las personas que se quieren incorporar al programa de doble doctorado. Y el día de hoy les quiero presentar a la doctora Cintia Fernández, que está acá, que ella es una de nuestras graduadas del programa de doble grado, pero además tiene la particularidad de que su trayectoria fue una de las más exitosas, o ha sido una de las más exitosas en nuestro programa, porque fue nuestro primer y aún único, pero ojalá en el futuro haya más, nuestro primer cum laude en la Universidad de Groningen, en el programa de doble grado. En Países Bajos, un cum laude, que sería como una mención honorífica, solo se lo adquieren como el 5% de todos los doctorados de los Países Bajos. Entonces, básicamente es uno de los mejores doctorados que han habido en los Países Bajos. Entonces, y es una estudiante de la UNAM, entonces ella les va a contar todo cómo esto se realizó, les va a contar un poco de su investigación, de cómo se graduó, etcétera, etcétera. Entonces, pues los dejo con ella y de todos modos estaré pendiente de cualquier pregunta, ¿vale? Pues los dejo con ella.
Hola chicos, buenas tardes a todos. Primero que nada quiero agradecer al comité organizador por invitarme a formar parte de este evento y sobre todo por poderles compartir mi experiencia, que lo puedan oír de viva voz de casos de la vida real, ¿no? Bueno chicos, yo empecé mi formación en la Facultad de Química hace muchísimos años. Yo estudié Química Farmacéutico-Biológica en la Facultad de Química. Ahí tuve muchos amigos y pude hacer mi tesis y ahí obtuve mi título de QFB. Posteriormente, después de graduarme, me preguntaba yo qué hacer porque me gustaban muchísimas cosas, ¿no? Me gustaba la bioquímica en específico, me gustaba muchísimo, pero también me encantaron muchísimo los equipos de cromatografía que vimos en química analítica, ¿no? Por ejemplo. Entonces, ¿de qué manera podía yo hacer como un híbrido de esas dos cosas? Pues resulta que en mi servicio social llegué al Instituto Nacional de Pediatría, en donde justamente se manejan estas dos cosas. Y está muy enfocado al estudio de enfermedades raras, o sea, con niños que tienen enfermedades genéticas que causan un error innato del metabolismo, es decir, alteraciones en el metabolismo, que entonces hacen que se acumulen metabolitos en sangre, y esos metabolitos en sangre se cuantifican con técnicas de cromatografía y de espectrometría de masas. Entonces, para mí fue así como, pues de aquí soy, ¿no? Entonces allí pues hice mi servicio social, estuve muy contenta, y entonces en ese mismo instituto se presentó la oportunidad de que yo hiciera mi maestría en ciencias biológicas con un doctor que nada tenía que ver con errores innatos del metabolismo, ¿no? Pero es que durante mi estancia en el instituto también asistí a seminarios y entonces me empapé de muchos otros temas que había ahí en el instituto, y entre esos estaba el estudio de proteínas metabólicas del parásito intestinal Giardia lamblia, ¿no? Que es muy, muy estudiado y que además el profesor con el que empecé la maestría es timólogo, o sea, es de los de la vieja escuela de la triosa fosfatoisomerasa, que es pura bioquímica básica, ¿no? Padrísima también, cinética enzimática. Entonces a mí se me abrió otro mundo, ¿no? Tremendo. O sea, ahora tengo las vías metabólicas, los metabolitos, la cromatografía, la espectrometría de masas. A mí el cerebro ya me explotaba con eso, ¿no? Y en cuanto veo ahora que se pueden estudiar las proteínas, ¿no? A nivel de estructura y función, y que yo puedo hacer mutantes y que puedo hacer proteínas recombinantes y que, o sea, entré a todo otro mundo que dije, no, a ver, espérame, para poder estudiar los errores innatos del metabolismo necesito aprender esto otro, ¿no? Pero desafortunadamente en el laboratorio donde yo hice la tesis no se especializaban mucho en la estructura y función de proteínas, ¿no? Nos dedicamos más a estudiar a los pacientes, a diagnosticarlos, a seguirlos, a toda la cuestión analítica del laboratorio clínico, ¿no? Pero para toda la parte de investigación de esto de, a ver, ¿por qué esta proteína, ¿no? Que está mutada en este aminoácido en específico, ¿por qué da este cuadro clínico a este niño que estoy conociendo, ¿no? Entonces veía yo venir un mundo así inmenso de posibilidades de investigación de todo este nicho de oportunidad. ¿Por qué las proteínas que tienen una variante patogénica, se llama, ¿no? ¿Por qué dan un cuadro clínico, ¿no? ¿Por qué se traducen en una enfermedad y en algunas veces una enfermedad menos severa que en otros pacientes, ¿no? Entonces, pues ahí también volví a hacer clic y decidí irme a estudiar la maestría con el doctor Gabriel López Velázquez, y empezamos a trabajar a la triosa fosfatoisomerasa de Giardia lamblia, y empecé a jugar haciendo mutaciones y midiendo actividad enzimática de TIM y ver qué le pasaba. Ellos lo que estaban buscando era cómo matar a Giardia, ¿no? Porque saben que este parásito pues es cosmopolita, se encuentra en toda la población, pero además los tratamientos de primera línea para este parásito ya han desarrollado resistencia, ¿no? Como sabemos que la cuestión de resistencia a antibióticos está tremenda, ¿no? Si no acabamos los tratamientos, lo único que estamos haciendo es hacer más resistentes a esos microbios, ¿no? Entonces, por eso han surgido cepas que ya son resistentes, y la necesidad de encontrar terapias alternativas también para acabar con Giardia son aún vigentes, ¿no? Se necesita porque existen las cepas resistentes. Y nosotros estábamos pues tratando de encontrar una molécula que le diera, ¿no?, a la TIM para proponerla como un nuevo fármaco, que ya también hay una historia de hace muchos años, ¿no?, acerca de eso, pero mi tutor fue de los primeros que propuso al omeprazol y a todos los prazoles y esas moléculas derivadas del omeprazol como posibles antigiardiásicos, y además que le pegan a la TIM, ¿no?, a la triosofosfatoisomerasa, entre otras enzimas, ¿no? Bueno, en ese andar de la maestría resulta que mi tutor, el doctor Gabriel López Velázquez, es muy amigo del doctor Hugo Luján en Argentina, en Córdoba, en donde es uno de los centros más especializados en Giardia lamblia. El profesor Luján ha publicado muchísimos artículos desde la vieja guardia de Giardia lamblia, muchísimos conectes en Estados Unidos, y nos invita a hacer una estancia de 3 meses en su laboratorio. Y pues vámonos, ¿no? Allá, ahí la foto que les muestro es de donde estoy yo trabajando en una campana de extracción con Yardia, y pues estábamos bien contentos de estar ahí, ¿no? En ese laboratorio que también tiene muchísima tecnología y son de los más top en el estudio de Yardia lamblia. Bueno, entonces ya que hice ahí mi investigación y que después me titulé de la maestría, justamente trabajando con la caracterización funcional y estructural de mutantes de cisteínas de la triosofosfato isomerasa, pues ahora ¿qué sigue, no? ¿Qué sigue? Fíjense que mi tutor no nada más se dedica a trabajar con la team de Yardia, sino que ahora se nos ocurrió, o sea, yo ya me quedé súper clavada con esto del metabolismo. ¿Qué pasa si ahora nos vamos a estudiar a la arginina deiminasa de Yardia, ¿no? No nada más nos quedemos con la triosofosfato isomerasa. ¿Qué pasa si yo le pego a otras enzimas que metabólicamente son súper importantes para Yardia, que son cruciales para su sobrevivencia? ¿Qué pasa con las cisteínas de esa enzima? ¿Qué pasa si yo le doy omeprazol, ¿no? Todo este tipo de cosas se nos ocurrían si yo le hago mutantes, ¿no? Entonces de ahí surgió mi proyecto de doctorado en el posgrado en Ciencias Biológicas de la UNAM, hablando sobre clonación, sobre expresión, purificación y caracterización de la enzima arginina deiminasa de Yardia. Y ahora lo que quisimos fue tratar de hacer un vínculo entre Yardia y los errores innatos. Es que estamos medio pirados, ¿no? A veces sí se nos gana la imaginación. Entonces dijimos, a ver, si la enzima arginasa humana, que es la responsable cuando hay variantes patogénicas de la enfermedad que se llama argininemia, ¿no? Porque se acumula la arginina, ¿qué pasa si yo genero un posible fármaco que pueda ayudar a que esa arginina se consuma? Y si esa enzima yo la saco de Yardia y se la doy a mi paciente en un tipo de cápsula, algo que haga que en el intestino mi paciente no le llegue tanta arginina que le causa daño, ¿no? O sea, el chiste es que tratando de hacer una posible tecnología de fármacos nueva, fue que surgió este programa, mi proyecto de doctorado. Sin embargo, nos topamos con un pequeñito detalle, que la enzima arginina deiminasa, uno de sus productos secundarios es el amonio. Y el amonio justo es el meollo del asunto en estas enfermedades del ciclo de la urea, que como no se puede generar urea para eliminar el nitrógeno, ¿no?, proteico, entonces se empieza a acumular el amonio y eso le causa muchísimo daño al cerebro. Entonces dijimos, no, pues estamos en un tremendo problema, ¿no? Porque no consideramos esa parte. Y bueno, le empezamos a dar un poquito de causalidad al proyecto. Dijimos, bueno, a lo mejor no sirve para argininemia, pero sí el hecho de caracterizar a esta enzima ADI de Yardia, que está muy poco estudiada, puede servir como para proponerla para como un nuevo blanco para generar un antiyardiásico, ¿no? Entonces estábamos en todo ese meollo. Y resulta que les platico que mi tutor, además de dedicarse a todas esas cuestiones metabólicas, también se dedica al estudio de las fibras extraídas de agave tequilana. Estas fibras son ricas en unas moléculas que se llaman fructanos. Los fructanos están considerados como los carbohidratos no digeribles, ¿no?, dentro del grupo de carbohidratos no digeribles. Estos fructanos tienen un efecto benéfico en el ser humano porque tienen un efecto prebiótico. ¿Eso qué quiere decir? Que los microorganismos de la microbiota intestinal se ven favorecidos con estos fructanos porque lo utilizan como fuente de carbohidratos. Nosotros los humanos no podemos digerir esos enlaces beta, pero ellos sí, y entonces a cambio producen ácidos grasos de cadena corta, y esos ácidos grasos de cadena corta son muy benéficos para la salud y están ampliamente estudiados con otras fibras dietéticas, otros carbohidratos no digeribles. Sin embargo, los fructanos de agave tequilana están muy poco explotados, y nosotros saben que México es el centro de diversidad de la especie de agave, ¿no? No nada más tequilana, hay muchísimas otras especies que están muy poco estudiadas. Entonces mi tutor junto con otros colaboradores ahí en el instituto hacen una serie de estudios clínicos en los que demuestran que estos fructanos del agave tienen un potente efecto prebiótico que además le da muchísimos beneficios a una serie de 600 niños neonatos que nacieron por vía, puede ser que hayan nacido vía vaginal o por cesárea, pero que muchos de ellos no tuvieron lactancia materna. Entonces lo que trataron de hacer fue suplementar a las fórmulas lácteas con estos fructanos y ver qué pasaba con la microbiota de los niños. Y resulta que cuando comparan las microbiotas, encuentran que la microbiota de los recién nacidos alimentados con suplementación de fructanos casi equipara al niño, al control, que sí tuvo siempre seno materno, ¿no? Lo cual no suena nada ilógico, puesto que la leche materna tiene estos HMOs o carbohidratos derivados de la leche, que son muy parecidos en molécula a los fructanos del agave, ¿no? Entonces ahí esos estudios fueron muy exitosos, empezaron mucho a buscar a mi tutor por esa cuestión de que este hallazgo estuvo muy bonito, y pues resulta que hay un doctor en Groningen, el doctor Paul de Vos, él se dedica, es especialista en estudiar fructanos de la planta de la achicoria. Esa planta de la achicoria es muy famosa en Europa, es muy utilizada como fuente de fibra, justamente porque ya se sabe de su efecto prebiótico. Incluso en Europa, por norma, se incluyen en los alimentos procesados como los cereales, tienen que llevar cierto porcentaje de fructanos o de fibra dietética como fuente de fibra. Y esto es porque se ha visto de los beneficios que tiene, ¿no?, el consumo de fibras en el ser humano en general. Y se ha visto también que en nuestra última era tecnológica y tan moderna, y nos ha cambiado los hábitos alimenticios, de tal manera que ahora pues yo pido, ¿no?, en Uber Eats una hamburguesa, mañana pido, ¿no?, de Kentucky, y entonces estoy olvidando y dejando de lado cada vez más los pobres vegetales que son muy benéficos para la salud. ¿Qué es lo que se trata de hacer con este tipo de normas? Pues que por lo menos la gente sí las consuma de alguna manera, ¿no? Porque se ha visto que previenen cáncer, previenen obesidad, previenen diabetes, son muy benéficas estas fibras. Entonces el doctor de Vos, que trabajaba muchísimo con estas fibras de la achicoria, ve los artículos del doctor Gabriel y dice, no, yo de ahí soy, ¿no? Necesito caracterizar esas fibras, quiero trabajar con ellas. El doctor de Vos se viene hasta México específicamente a buscar a mi tutor, así de, oye, yo necesito que hagamos una colaboración porque tus fructanos se ve que están buenísimos, ¿no? Y entonces ahí es como surge esa primera unión, digamos, UNAM-Groningen, ¿no? ¿Qué hacemos? ¿Qué es lo que podemos proponer? Pues empiezan a trabajar en proyectos, cómo trabajar en conjunto, y resulta que pues se requiere siempre de un estudiante, ¿no?, que lleve a cabo las cosas para que todo vaya marchando bien y para que dé beneficios. Entonces mi tutor me dice, a ver, yo creo que tú puedes con todo, ¿no? Ya saben, me hizo el coco wash. O sea, tú estás haciendo, ¿no?, Adi de Yardia, muy bonito, te está yendo muy bien, pero pues yo creo que también te puedes echar los fructanos, ¿no? Y así, ay. Pues sí, la verdad es que la idea sí me encantó porque a mí o sea, soy científica nata, ¿no? O sea, me dices, vamos a estudiar esto y a mí me emociona. Y yo a lo mejor mucho en mi cuestión de inmadurez, ¿no?, y de decir, pues va, ¿no?, sin saber hasta dónde me estaba metiendo, ¿no? Pues ya muchísimas cosas pendientes, aparte de mi proyecto de doctorado. Digo, pues va, pues vamos a meter el proyecto, si sale y se apoyan, pues órale, ¿no? Pues ya lo trabajamos y todo lo planteamos de tal manera que entonces, a ver, déjame ver si es la que sigue. Lo planteamos de tal manera que sí, pues dijimos, a ver qué pasa con los fructanos del agave. Y entonces ahí va Cintia, ¿no?, de México a Holanda. Llegué yo a Groningen en mayo de 2019. Resulta que surgió este programa, no sé si ya tenía tiempo, pero lo que me dicen es que en mi posgrado de Ciencias Biológicas donde yo estaba, yo iba a ser la madrina del programa, porque era la primera estudiante del posgrado de Ciencias Biológicas que se iba en esta cuestión de doble doctorado, ¿no? Entonces, bueno, pues había por ahí una ventaja. Dije, no le veía mucho el caso a hacer doctor, ¿no? Como que doble doctorado, pero lo que más me llamaba la atención era poder ir a visitar un lugar de primer mundo, ¿no?, en donde no te limita nada más que tu imaginación, porque presupuesto tienen a manos llenas, equipos hay de lo que se te ocurra, ¿no? Nada más la cuestión es saber bien qué es lo que quieres hacer, cómo lo vas a hacer, sobre todo para cumplir con los tiempos, ¿no? Porque nada más tenía yo 2 años. O sea, en este programa tus primeros 2 años los estudias del doctorado en la UNAM y luego te vas los 2 años restantes a Groningen, ¿no? Entonces, pues no tenía ya mucho tiempo y pues ahí llegué a Groningen, ¿no? Entonces, mi proyecto fue estudiar los efectos benéficos de los fructanos derivados del agave tequilana y esto fue que nos fuéramos a estudiar más allá del efecto prebiótico que ya está muy sobado, ¿no? Ya todo el mundo quiere saber, ¿no? Y ya se sabe. Pero ¿qué pasa antes? O sea, antes del colon tenemos al intestino delgado, ¿no? Y es mucho más largo que el colon, ¿no? Y entonces, ¿qué tal que hay un efecto directo de los fructanos con el epitelio intestinal? Que ya después de refilón lleguen al colon y hagan su efecto prebiótico muy padre, pero ¿qué está pasando en el intestino delgado, ¿no? Los fructanos podrán interactuar directamente con el epitelio intestinal, prenderán alguna señal, tendrán algún efecto en el sistema inmune del intestino, que es de los más importantes del organismo. Entonces, pues todas esas preguntas yo fui enviada especial a responderlas, ¿no? Entonces, fíjense que sí, llegué a instalaciones de primer mundo, padrísimo, todos los laboratorios gigantes, equipos a más no poder, llegas a unos almacenes que tienes tu credencial para abrir, y para el estudiante PhD en Groningen, el instituto está abierto 24/7, ¿no? No hay días feriados, no hay nada de eso. Entonces tú llegas y tienes a manos llenas todo lo que se te ocurra y lo que tú quieras, ¿no? Y tienes muchos compañeritos que ya pues están ahí de múltiples nacionalidades, en su mayoría chinos, que son una competencia tremenda. O sea, los chinos de verdad, mis respetos. O sea, esos chicos, yo pasaba un domingo en la mañana y ahí estaban sus bicicletas afuera, de que ahí estaban, ¿no? Yo podía pasar un domingo en la noche y ahí seguían. Esos chicos tienen una presión tremenda porque al ser tantos, quieren regresar a su país con un PhD en mano. Pues sí, mi chavo, pero atrás de ti vienen otros 1000, ¿no? Entonces aquí, para que yo te pueda dar una posición de regreso en tu país, pues nos vamos a los papers, la calidad de los papers, de qué impacto fueron, ¿no? De cuántos papers fue tu tesis. O sea, ya tú te mides por de cuántos capítulos fue tu tesis. Capítulo por capítulo es artículo publicado, ¿no? Entonces estos chicos pues estaban en una competencia tremenda, y creo que no nada más pasa en Groningen, en Estados Unidos también se sabe muchísimo, tienen fama los chinos de ser los más trabajadores, ¿no? Y sí, mis respetos para esos compañeros. Pues aquí están mis amiguitas, la que les estaba platicando, ¿no? Hice muchos amiguitos, establecí, además eso es bien importante, ¿no? Que tú veas no nada más en el punto en el que estás presente, sino que siempre vas a ir dejando, ¿no? En tu camino, huellitas que te van a después ayudar. O sea, que vas a establecer colaboraciones con gente que en el futuro va a ser un PI, que es un Principal Investigator en otro lado, ¿no? Que te llevaste de a cuartos con él durante sus estudios, pero que después pueden hacer colaboraciones muy bonitas. Entonces, también yo lo que les quiero compartir de mi experiencia es que ustedes, si se animan, ¿no? Vayan con mucha humildad, vayan siempre prestos a ayudar. Eso les va a servir en la vida en general, ¿no? No se cierren, no se aíslen. Siempre el trabajo en equipo es lo mejor que les puede pasar, porque algún día alguien puede necesitar de ustedes, pero algún día ustedes pueden necesitar de alguien, ¿no? Entonces siempre no se les olvide que siempre vamos dejando, ¿no? Huellas a nuestro paso. Miren qué altos son los holandeses. O sea, ven a ese chico que tiene que hacerle así a la puerta porque no cabe. En los Países Bajos, donde todos son altos, ¿no? Fíjense que además, no nada más conforme con ir a estudiar el doctorado, allá los estudiantes de doctorado tenemos la oportunidad de dirigir estudiantes, no nada más de licenciatura. Allá las maestrías son muy particulares porque los chicos hacen 2 semestres en la escuela tomando clases, laboratorios, pero luego los otros 2 semestres, uno de ellos se van a la industria, si así lo eligen, ¿no? Porque allá sí hay industria que pague bien a los posgrados, ¿no? Posgraduados y a los becarios. Entonces se van y hacen estancias de un semestre en alguna industria, o también pueden hacer estancias en los centros de investigación, como fue el caso de Mirte, ¿no? Ella decidió hacer sus 2 internados, que les llaman, en los laboratorios de investigación, y entonces a mí me llegó pues esta chica muy dispuesta a ayudar, trabajamos súper bien, codo a codo. me ayudó muchísimo, yo le enseñé muchas cosas, aprendí muchísimas otras de ella, ¿no? Sobre todo de la cultura, de cómo son de organizados. Los holandeses son a veces a rayar en la obsesión, ¿no? O sea, yo por ella ahora traigo un cuadernito así de mi agenda, de que hora a hora qué voy a hacer, ¿no? Paso a paso para la semana siguiente que tengo, y de verdad, o sea, así como aprendí yo de ella aprendió muchísimas cosas de mí, ¿no? Yo ya traía pues toda la historia que les he platicado, y entonces no nada más a ella, sino a todos mis compañeritos con los que yo estaba, pues les enseñé muchísimas cosas y aprendí mucho de todos ellos. Y además de traerme pues un PhD en la mano, me traje un estudiante de maestría, ¿no? En mi currículum. Pero bueno, no todo fue trabajo, chicos. O sea, estando en Europa, pues ni modo que en el verano que me voy a quedar a hacer ahí, pues a sentarme, pues. Además, Londres estaba a tiro de piedra de Groningen. O sea, literal es un vuelo de 1 hora que cuesta menos como 100 euros, por decirte, ¿no? Entonces, pues me fui el verano del 2019 a Londres, también anduve ahí paseando con amiguitos, le llamamos el Latin Squad, porque pues ahí nos conocimos de Colombia, ¿no? De muchos lados latinos. Y pues fue muy padre experiencia en todos los sentidos. También en la cuestión cultural, ¿no? Desde aprender a usar la bici otra vez, porque yo desde que tenía como 12 años, pues ya no la volví a usar, ¿no? Pero allá en Países Bajos, pues todo el tiempo es bici, ¿no? Ya yo después para ir a la esquina no dejaba la bici, ¿no? Para ir al mercado. Es muy padre porque además, como ese país es muy plano y la cultura del ciclismo está tremenda, ¿no? O sea, casi prácticamente todo es bicicleta, ¿no? Muy pocos autos, pero sí, además de que las ciclovías son ciclovías verdaderas, ¿no? También, porque cuando ya regresé, pues, híjole, qué decepción, yo sí me deprimí porque yo salía con mi humilde bici y pues, ¿cuál, no? Pasan y sí, o si no te la mientan, te avientan el carro aquí, ¿no? Digo, para que también tomemos en cuenta como automovilistas que los ciclistas tienen mucha importancia, tienen mucha prioridad, ¿no? Ya sé que la principal prioridad es el peatón, pero luego sigue el ciclista, ¿no? Y si todos tuviéramos una mejor cultura del ciclismo, reduciríamos muchísimo los niveles de contaminación de nuestra ciudad, ¿no? Esas son de las lecciones que yo también me traje de allá. O sea, ¿por qué en México no podemos ser más de una cultura así, ¿no? Del ciclismo, de respetarnos más, de ayudarnos más. Yo he metido ya al gobierno de la ciudad, sí, por favor, más ciclovías, pero ciclovías de verdad, no pintaditos ahí que pues, nadie respeta, ¿no? De las cosas más bonitas que me traje y que más nostalgia me dan es la bicicleta. Lo que más extraño es la bicicleta, porque ya solamente en fines de semana puedo venir aquí al circuito, ¿no? A dar unas vueltas o algún parque, pero pues nada más. Pero pues luego llegó la pandemia, chicos. Llegó la pandemia y entonces pues todo se volvió lo que ya sabemos, ¿no? El Zoom, el Teams, ¿no? Todos mis tutoriales ya eran si de por sí eran así, porque pues la gente, mi comité tutoral estaba en México, pues ya después los seminarios también de allá del instituto se volvieron igual, ¿no? Pura cuestión de Zoom, pura cuestión online. Afortunadamente allá se organizaron súper bien, pues sí, no era de extrañarse, son muy organizados, y entonces nos pusieron en tandas a todos los que estábamos en el departamento para trabajar, para poder ir a trabajar al laboratorio, pero pues que no hubiera mucha más gente de la que no se podía por la cuestión de la distancia, ¿no? Ya saben todas las medidas, pero yo afortunadamente sí pude ir a hacer un híbrido entre ir a trabajar al laboratorio en la mañana, en la tarde, y después dedicarle mucho al trabajo de escritorio, ¿no? Y entonces, lo que les digo, pues ya no podía yo ir a ningún lado, ¿no? Ya, Londres fue debut y despedida. Estaba en muchísimos países de Europa, pero ya no se podía ir a ningún lado. ¿Qué hice? Pues me metí de lleno al laboratorio, ¿no? Que fue por lo que yo llegué y que estaba súper encantada. Pues ahora no me sacaban del laboratorio. En los turnos que se podía, cuando alguien no quería usar su lugar, yo ahí estaba, ¿no? Yo aquí vine a trabajar. Y como a mí me encanta el laboratorio, no me alcanzaban los pasillos, no me alcanzaban ¿no? Todo era mío. Y entonces, pues sí, también hice un poquito de home office. Pero ahora mi celular se volvió así, ¿no? Las fotos que les pongo ahí, ¿no? Cuando voy a mis fotos, toda esa época, así, puras fotos de pantallas, experimentos, ¿no? Porque pues a eso me dediqué. de tal manera que cuando mi estancia terminó en mayo de 2021, pues yo me regresé a México con un montón de resultados bajo el brazo, mismos que generaron una tesis de 7 artículos científicos publicados en revistas de alto impacto. Entonces, lo que más le sorprendió a la gente de allá y a la gente de acá es que yo en tan poco tiempo haya logrado hacer tantas cosas. Pues sí, pero también hubo una pandemia, ¿no? También me metí de lleno, no hacía otra cosa, ya les competí mucho a los chinos, ¿no? Pues era obvio. ¿Por qué? Porque el resultado lógico de algo que tú haces, ¿no? Pues es el resultado lógico. ¿Cuál es el resultado lógico del trabajo, del esfuerzo? Pues el éxito. No hay más, ¿no? Si trabajas duro, ¿qué te va a dar de resultado? Pues un éxito, ¿no? Entonces aquí está la portada de mi tesis. Además, me dice mi tutor allá en Groningen, el doctor Paul de Vos, oye, Cintia, salió esa de la Global Prebiotic Association. Pues mete uno de tus papers, a ver, tú mételo, ¿no? Bueno, pues lo metí, apliqué a la convocatoria y pues resulta que me gané el Young Researcher Grant Recipient de 2023 de Fundamental Research por uno de mis artículos de los del doctorado, ¿no? Pues son cosas que uno no se espera, ¿no? Pero es lo que les digo. O sea, el resultado lógico del trabajo diario es el éxito. No hay más, no hay otra receta mágica, no hay más, ¿no? Y más cuando te gusta, pues mucho más, ¿no? Bueno, después se vino la hora de la defensa. Es todo un tema, chicos. La defensa de allá es diferente a la de aquí. O sea, aquí estamos acostumbrados a que llegas, ¿no? Haces tu mini presentación ahí de 20 minutos, platicas lo que hiciste, está el jurado y te hacen preguntitas, ¿no? Como 10 minutos. No, allá no hay presentación, ya el jurado leyó la tesis. Entonces, llega, se cierra la puerta y son 45 minutos de tortura. Hasta que llega el alguacil, que le llaman, es el señor que va enfrente, que es el que maneja todo, y dice, hora finita. O sea, se acabaron las preguntas, se acabó el tormento, ya 45 minutos fueron suficientes de preguntas, ¿no? Yo tenía 6 jurados internacionales, o sea, me pusieron una doctora de Turquía, un doctor de Brasil, 2 mexicanos, y entonces muchos estaban en línea porque no pudieron ir ese día, ¿no? Fue apenas el 3 de mayo. Entonces, entre que no se entendía muy bien, se cortaba, la transmisión en algunos estuvo horrible, no les entendía bien a las preguntas, los nervios, la familia ahí, ¿no? Los amigos, bueno, pero finalmente, pues yo lo que rogaba era por salir viva de ahí y contestar adecuadamente, ¿no? A todas las preguntas. Sí, es un momento muy, muy bonito, pero también súper estresante, ¿no? Y ya, pues también se salen a deliberar los jueces. Tienes 2 amigos que son como tus damos de honor, que se llaman los paraninfos. Es muy ceremoniosa la cosa. Hagan de cuenta que entraron a Hogwarts, ¿no? Así el edificio está padrísimo, todo de madera, pues ahí lo ven, ¿no? El salón de los exámenes, todo muy lindo, muy ceremonioso el asunto, y ya después de que salen a deliberar, pues me dicen que sí tiene usted el grado, pero además se lleva el famoso cum laude, ¿no? Que eso es algo que no se lleva a cualquier persona, ¿no? Es lo que me estaban platicando, que además, sí, o sea, yo ni idea, ¿no? Porque además la tesis se manda primero, si se propone para cum laude, se manda primero con asesores o gente experta a nivel internacional que lo revisan, además que el día del examen tienes que hacer una excelente defensa, y no conforme con eso, todos los votos tienen que ser unánimes. O sea, si una persona dice no, a mí no me contestó bien, todo lo demás se cae y ya no hay cum laude, ¿no? Entonces sí se requiere de que sea todo voto unánime para que se pueda dar el reconocimiento. Y dije, ay, bueno. Ahí estoy con mi título, con mis 2 tutores. Y el embajador de México en Países Bajos dio un discurso felicitándome. Fue todo una notición ahí, porque soy de las pocas latinas que tiene este tipo de distinciones, entonces fue así como lo máximo. Yo todavía sigo sin creérmelo, ¿no? Es muy bonito, pero a mí más que las cuestiones estas de las medallas y eso, lo que a mí me nutrió muchísimo fue todo lo que aprendí, todo lo que yo pude dar de mí, ¿no? Todo lo que pude hacer de colaboraciones, ahora con Paul de Vos seguimos trabajando, pues ahora ya tengo una plaza de investigadora en ciencias médicas ahí en el instituto, y además soy profesora ya de asignatura en la Facultad de Ciencias, porque a mí lo que me apasiona, además de la investigación, es la docencia, ¿no? Me encanta explicar, compartir experiencias, como le llamamos una amiga y yo, ¿no? En los cálculos, así llegar hasta las últimas consecuencias a por qué este mol con este mol dio este número, así obsesivo, ¿no? Pero a mí me fascina muchísimo enseñar y compartir para todos. Entonces yo les quiero dar unos pequeños consejos para los que se animen a hacer el doble doctorado o el doctorado por allá. El inglés, chicos, o sea, es para ayer. El inglés hablado es para ayer, ya. O sea, tienen que ponerse súper duchos y les voy a decir por qué. Porque en cuanto te topas a la primera persona allá y empiezas con I want, ¿sabes qué? A los 2 segundos se da la vuelta y ya no te vuelve a hablar. Porque la gente no tiene tiempo para estar lidiando con gente que no puede hablar inglés adecuadamente. Es muy triste, suena muy feo, es agresivo, pero es la realidad. O sea, yo además de mis tiempos libres, que me quedaban pocos, le ayudaba a mis amiguitos chinos a practicar, porque los pobres tienen, pues por su lenguaje, ¿no? Su lengua nativa, tienen muchísimas complicaciones para hablar inglés y aparte los nervios que te dan de que estás en un lugar extranjero, ¿no? Todo se te conjunta y entonces para hilar una oración se vuelve bien difícil. Entonces, pues ahí organizábamos los clubs de conversación con mis amiguitos para poder practicar entre todos, ¿no? Yo aquí hice el curso del TOEFL en el CELE, que es muy bueno, se los recomiendo. Y si les van a pedir aparte 600 puntos mínimo de TOEFL para poder aplicar al doble doctorado. Entonces, pues ya, el inglés es para ayer, por eso les digo. Sean disruptivos, chicos. ¿Qué quiere decir ser disruptivo? Es pasar las barreras que existen, imaginarias o verdaderas, ¿no? Es el decir, ¿yo por qué no? O sea, no se pongan limitaciones, eso quiere decir ser disruptivo. Sálganse de lo común, siempre apuéstenle, hagan su mayor esfuerzo y nunca se den por vencidos antes de dar la batalla. Y sí se puede, sí se puede. Es trabajo duro, sí, es rudo, sí. Se pasan tiempos de soledad, sí, ¿no? Los mexicanos somos muy apapachadores y muy familiares y muy de nuestra comida, ¿no? Y muy de nuestra cultura, pero sí se puede. O sea, si se tiene buena voluntad, ganas de aprender, ¿no? Ganas de ir a conocer cosas nuevas, por supuesto que se puede. Y lo que les vengo diciendo, ¿no? Desde que empecé, el éxito es el resultado lógico del trabajo constante, no hay más. Eso sí, las buenas relaciones con sus tutores, chicos, eso también. O sea, traten de conseguir tutores con los que hagan clic, con los que se lleven bien, ¿no? Que sea gente que además de ver la cuestión académica, vea la cuestión humana. No muchos doctores lo tienen, ¿no? Y en especial gente de países nórdicos como en Holanda, sí tiene algunas culturas diferentes, ¿no? Yo tuve la fortuna de que el doctor Paul de Vos es un excelente ser humano, primero que nada, ¿no? O sea, teníamos los que le llaman el face-to-face meeting, o sea, todo el one-to-one, algo así. Todas las semanas, ¿no? Se tienen entrevistas con el tutor para ver cómo vas, ¿no? Le llevas tus resultados, le presentas algo. Pero antes de que empezáramos a platicar de trabajo, era, ¿cómo estás tú? How's life? O sea, tú, Cintia, ¿cómo te sientes? Ya después platicamos de lo demás. ¿Estás pasando por alguna dificultad? ¿Has tenido algún problema? ¿Cómo te sientes? ¿No? Siempre verificando la parte humana de la persona. Porque claro, para que tú puedas dar el 100, pues primero como humano uno tiene que tener satisfechas ciertas necesidades, ¿no? Fíjense que yo llegué, allá en Groningen hay una crisis de vivienda tremenda, porque es una ciudad muy estudiantil, todo mundo necesita un cuarto donde vivir, ¿no? Y yo dije, pues no me voy a arriesgar a pagar algo aquí que después capaz que me hicieron fraude, ¿no? Mejor llego a un hotelito, ¿no? Mis papás con mucho esfuerzo, y luego ya voy viendo dónde hay lugares, dónde hay casas, qué cosas habrá, ¿no? Entonces era caro el hotel en donde yo estaba y mi supervisor me dice, ¿dónde te estás quedando, no? Cuando llegué. Digo, en el Student Hotel. No, pero así se puso como, porque además son codos lo que le sigue. No, estás pagando muchísimo, pero además de que su personalidad es así, ¿no? Él es muy aprensivo. No, a ver, ahorita. todos vamos a ponernos a buscarle a Silvia dónde vivir, porque no puede ser, está en un hotel muy caro, no. Y entonces todo el mundo me ayudó y uno de los compañeros chinos conocía a una casera, una señora china que tenía una casa que rentaba los cuartos para los estudiantes, pues ahí fue y me presentó, habló en chino por mí, con la señora, porque ella no hablaba inglés. Aquí está mi amiga, por favor, déjele quedarse en un cuarto. Y por ese chavo fue que me dieron la oportunidad de estar en ese cuarto, porque pues era comunidad china muy cerrada, ¿no? Primero, pues los compatriotas, no, finísimas personas. Yo me llevo experiencias padrísimas de todas esas gentes, y todo porque mi tutor, pues ahí, ¿no? Contribuyó muchísimo a eso. Y además ese es el último tip, chicos. No nada más, pues qué padre que sí sea muy buena persona y, ¿no? Pero estamos en un mundo bien competitivo y en el que nosotros vamos por un objetivo bien claro. Y eso no se los dicen muchas personas, pero ustedes PubMeden, ¿no? En lugar de googleen, PubMeden a los tutores. O sea, vean cómo publican, qué tanto publican, cuántos artículos, porque entonces ustedes no se trata de que, más bien se trata de que se suban a un tren que va andando, no de que vayan empujándolo atrás, ¿no? Para que camine. O sea, también hay que ver a dónde nos vamos a ensamblar, ¿no? Con alguien que ya tiene el caminito andado, que tiene la experiencia, que buenísimos para publicar, o sea, mi tutor así de la nada, a ver, ahorita vamos a hacer un review, ¿no? Con eso vas a empezar, vas a hacer una revisión de la literatura, de todas maneras te tienes que poner a estudiar, ¿no? Para que veas en qué hemos hecho con las fibras de achicoria, cómo vas a ensamblar las fibras del agave, en dónde las vas a montar, qué vas a estudiar, ¿no? Pues ya que vas a leer, pues hazte una revisión, ¿no? No te cuesta nada, ya lo vas a leer. Ahí acomódate una revisión. Y ese va a ser el capítulo 1 de tu tesis, una revisión de la literatura. Luego ya, conforme van dando resultados tus experimentos, vamos armando las publicaciones, ¿no? Entonces, alguien que tiene la visión, ¿no? De llevarte a buen puerto, de llevarte a buen destino, es súper conveniente. Entonces, también tomen mucho en cuenta eso cuando busquen las colaboraciones, que sea una colaboración bien establecida aquí entre México, ¿no? Y allá, bien ensamblada. Sobre todo que se vayan bien definido su convenio, porque aquí en la UNAM se hace un convenio personalizado por estudiante en el que diga, el encargado es el supervisor tal en México, el otro doctor allá en Groningen, va a haber tantas publicaciones de por medio, ¿no? Después no me salgan con que ahora se les ocurrieron 2 más, porque además el tiempo apremia, ¿no? O sea, uno va muy bien a lo que va. Entonces, ustedes bien abusados para que pues tengan una historia de éxito. Y pues ya, ahí está mi Hogwarts. Ese es el edificio científico, el académico.
Muchísimas gracias por tu amena presentación. Estoy segura que fue inspiradora o está siendo inspiradora para muchos. Así que chicos, no tengan miedo, anímense. Tenemos tiempo para algunas preguntas. ¿Alguien quiere comentar algo, alguna curiosidad? ¿Algo más que quieran saber? Tenemos varias minutos.
Bueno, ¿qué sucedió con tu trabajo con Yardia Lambia?
Bueno, fíjense que yo estudié los TLRs, estos receptores inmunes en el intestino. Ahí fue donde ensamblé el trabajo de los fructanos del agave. Y luego estando ahí se me ocurrió, pues ¿por qué no veo qué pasa con la ADI de Yardia, esta proteína que yo estaba estudiando? No se sabe nada, si activa TLRs, cuál, ¿no? Cómo, es dosis dependiente. Como todos mis trabajos fueron in vitro y yo ya traía la experiencia de sobreexpresar y purificar a la proteína, pues me llevé el gen en un tubito, así en mi equipaje, y allá monté otra vez la sobreexpresión y la purificación. Entonces andaba yo en 2 negocios, ¿no? Le llamaba yo ADI Story y Fructans Story, ¿no? Todos mis archivos así se llaman porque todo lo que hice de los fructanos del agave aproveché y también lo hice para ADI, entonces me traje doble trabajo.
Muy, muy bueno.
Hola.
Hola.
Primero que nada, muchas felicidades. Creo que puedo decir, bueno, no sé si por todos, pero eres un ejemplo a seguir. La admiración es, creo que en general, no solo en la cuestión académica, sino yo creo que también en la cuestión personal. porque yo sé que ir a otro país es muy difícil, no solamente en la cuestión académica, sino todo lo que pasa alrededor de tu vida diaria, la salud mental es muy importante y más que nada con la pandemia, creo que fue un poquito más difícil, pero la verdad, en verdad te quiero felicitar porque eres, no sé, eres una inspiración, ¿no? Una inspiración. Y te quería preguntar dos cosas. Una es, no sé si la cuestión del doctorado a ti, ya sea aquí en la UNAM o si no allá en Groningen, promuevan la divulgación, o sea, en cuestión de ir a conferencias, no ir a conferencias. Yo sé que fue la pandemia, pero pues no sé si en primer lugar como que en el contrato diga sí hacer como conferencias.
En el convenio no viene como tal, pero sí, mi tutor, ya tenía yo pagado antes de la pandemia la estancia para ir a Turquía, porque se hace cada 2 años o cada año este simposio de fructanos en general, ¿no? Y entonces es mundial. Hay una asociación que estudia los fructanos a la que pertenece mi tutor, que de veras me dijo que a él le toca organizar el siguiente, y entonces va a ser en Groningen. Y entonces ya estaba todo, yo ya tenía mi resumen, ya lo había metido, me lo habían aceptado para el congreso, pero pues se vino la pandemia y ya no pude ir al congreso, ¿no? Ya no se hizo. Y sí, o sea, hay muchísimos lugares donde tú puedes divulgar, hay muchísimos espacios para los estudiantes de doctorado. Me acuerdo ahorita de un concurso que se llama Describe tu proyecto en 2 minutos, ¿no? Entonces así de, a ver quién logra decir, ¿no? Todo lo que se dedica en cuestión de divulgación, nada más en 2 minutos y que te entienda la gente, ¿no?
Sí.
O sea, sí, hay mucho.
Ok. Y la segunda es, ¿tú obtuviste 2 títulos? O sea, tanto aquí, bueno, allá, tanto acá.
Sí.
¿Hiciste me explicaste tu tesis 2 veces?
Vengo llegando de Groningen, ¿no? Hace 2 meses.
Sí.
Con título en mano, eso es lo padre, ¿no? O sea, el título te lo entregan en ese momento. Y ahorita aquí estoy en trámites administrativos. Esa misma tesis nada más le tuve que traducir introducción y discusión. Es otro jurado nuevo, ¿no? La tiene que volver a revisar y tengo que pasar por todo el trámite de titulación aquí en México, el examen, ¿no? Profesional, todo. Y entonces voy a tener el título de la UNAM. Entonces yo tengo doctorado por la Universidad de Groningen y por la UNAM.
Genial. Ok, vale. Gracias.
Pues primero felicitarte y la segunda es si es doctorado tradicional o directo. Digo, los chicos que están de licenciatura pueden pensar en la posibilidad de irse directo de la licenciatura.
Las dos, pueden ser las dos.
¿Hay las dos? Ok, muchas gracias.
Yo tenía maestría, pero también puedes hacerlo directo.
Ok, muchas gracias.
Hola y felicidades.
Gracias.
Bueno, mi pregunta va más que nada, tú una vez acabando la licenciatura, ¿tú te llegaste a pensar en seguir en la parte científica o ya tenías planeado también trabajar si no se te daba?
No, la parte científica para mí siempre fue, o sea, yo dije no, investigación, o sea, yo veía a mis amiguitos de la carrera, ¿no? De química, los químicos en alimentos, los QFBs, muchos se fueron a la industria y hacían muchísimas cosas de regulación, pero para mi modo de ser era muy aburrido porque ya se te acabó, ¿no? Es lo mismo siempre y a mí me encantan los retos de todos los días, ¿no? Algo por resolver diario, o sea, soy muy hiperactiva, ya me vieron, muy acelerada, entonces yo no puedo estar quieta en un solo lugar. Lo mío siempre fue, no, investigación.
O sea, saliendo tú te abriste paso para la investigación.
Me encaminé hacia la investigación. Ajá.
Yeah. Gracias.
Hola, muchas felicidades por todos tus logros y tus éxitos. Quisiera preguntar, pero bueno, no sé si sabes al respecto de, en el doctorado, pero para postdoc también existen posibilidades en Groningen, o es más complicado al respecto de los postdoc, ¿qué sabes?
Yo te puedo platicar de los compañeros que conocí y a lo mejor Andrés nos puede ampliar un poquito más. Yo tuve varios compañeros que acabaron el doctorado y consiguieron postdoc allí mismo en el mismo instituto, en el Centro Médico Universitario de Groningen. Lo que sí sé es que no los dejan ser postdoc del mismo doctor con el que hiciste el doctorado, al menos el primer postdoc, ¿no? O sea, sí se tuvieron que ir a buscar a otro lado y luego regresaron a hacer un segundo postdoc ahí mismo donde se doctoraron. Son de 2 años, y ya. Ah, perdón, Andrés.
Sí, sí. Nada más, o sea, como para también aclarar, sí, o sea, definitivamente podría decirse que es un poco más complicado porque digamos que el esquema de doble grado que tenemos ya está bastante cimentado. Voy a hablar más de esa parte técnica el jueves, pero o sea, como de cuánto les dan de beca, cómo se financia, cuáles son los requisitos específicos y demás. Entonces, pero de todos modos, básicamente para que ustedes más o menos tengan una idea, para los estudiantes de doctorado ya la UNAM y Groningen tienen una beca establecida, hay una forma en la que se financia este asunto, pero para los postdocs no es tan fácil porque o lo paga directamente la Universidad de Groningen, o sea, por ejemplo, si los recibe Groningen, lo tendría que pagar el departamento, o ustedes tienen que aportar su beca, por ejemplo, con CONACYT, ¿no? Entonces son lugares mucho más escasos, tienen que buscar un departamento que les quiera contratar y demás, entonces pues se vuelve un poco más complicado, pero bueno, o sea, no significa que no sea posible, solamente es como cuestión de buscar, entonces sí es ligeramente más complicado, pero sí, nada más para complementar.
Mi pregunta es sobre qué programas de doctorado de la UNAM participan en este convenio y otra, bueno, entendí que la doctora Cintia obtuvo pues esta oportunidad sobre la marcha ya con su doctorado iniciado. Ahora, una vez al entrar al doctorado, ¿ya se tiene que considerar esta oportunidad de doble grado o igual se puede sobre la marcha ya iniciar un proyecto?
Ok, sí. Todos los programas de la UNAM pueden participar, no todos han participado y todos tienen sus propias reglas y son diferentes en muchas cosas. Para ustedes que probablemente varios o varias tienen antecedentes de química y si quieren entrar al programa de ciencias químicas, sí es muy importante que desde el principio hagan saber que tienen la intención del doble grado. Si no, o sea, en otros programas hay otros programas que son más flexibles y se puede hacer sobre la marcha sin duda, pero mientras lo hagan antes del cuarto semestre a lo mucho, ¿no? O sea, siempre entre antes vean la colaboración, es más fácil para nosotros establecer todo el caminito que se va a hacer hacia el futuro. Entonces, sí, definitivamente todos pueden ser y dependiendo del programa es qué tan estrictos son con los tiempos, ¿no? Pero eso es como detalles.
Ok, gracias. ¿Y los requisitos se pueden consultar en alguna página?
Sí, los requisitos muy generales están en... Sí, bueno, o sea, rápidamente, o sea, el jueves les doy con más detalles todo esto, pero si quieren meterse rápidamente o darle un primer vistazo, www.rug.nl/UNAM. Ahí hay información muy general y ya yo los invito a que el jueves vayan a preguntarme con más detalle, ¿vale? Porque creo que ahorita ya estamos sobre el tiempo, ¿va?
Gracias.
No, de nada.
Bueno, pues muchas gracias a todos por su asistencia. Muchas felicidades, Cintia, por esta súper plática muy interesante y motivadora para todos y todas aquí. Nos vemos en las siguientes sesiones en la tarde. Tenemos la clase del doctor Heller Santos, no se la pierdan. Y con ella vamos a empezar la siguiente parte del día. Muchas gracias. Y para todos los que quieran saber más detalles sobre los procesos para hacer alguna actividad en la Universidad de Groningen, especialmente de doble grado, acompáñenos el próximo jueves. Ahí vamos a volver a platicar con más detalle de estos aspectos. Muchas gracias.